Yep. Okay. Now that I'm unmuted. Thank you again, virtual and in person for coming, despite the weather we've had, the fun weather we've had here in East Texas. As interim president of FlowTex, it is my honor to open the 2020 FlowTex meeting. Thank you one and all that have made the effort to be here in person as well as virtually. FlowTex is an associated society with ISAC, the International Society for the Advancement of Cytometry, and FlowTex was founded by Joel Cederstrom in 2007, and little did we know that FlowTex would continue to grow into what it has become today, with this event being our 15th annual conference. Quite an accomplishment. In the last in-person meeting we had, February of 2020, we had that one, and then immediately following that, the pandemic hit, which shut us down. As many of you know, the 2021 meeting was virtual and is up on FlowTex's YouTube channel as we speak, and you can still visit that one. That was a very successful meeting, and I'll come around to that particular meeting shortly. However, when 2022 rolled around, the pandemic showed signs of easing. We were considering running FlowTex 2020 virtual again. To be candid, yes, we were all in a Zoom meeting, planning another meeting to have a virtual meeting, and none of us were really enthusiastic about another virtual meeting. We were, as many of you were, zoomed out. We were tired of Zoom. We lost count how many webinars and half-day conferences we had signed up for, but barely had attended 45 minutes to an hour's worth before a distraction took us off of our monitors. I'm not sure, but I think it was Joel who asked, yes, in a Zoom meeting, you think we can try to run 2020 for real? Run it in person? And we went around the horn and agreed, let's try this. Let's see if we can make this happen. But with the pandemic showing signs of waning just a bit, the big question in capital letters was where to have it. Our previous venue was not having in-person meetings. Other venues, both academic and non-academic, either limited us to no more than 30 people in total attendance or wanted an astronomical sum of money. A committee member, Bill Maritoja, inquired here at the University of Texas Institute of Molecular Medicine, where we are now, about having the meeting here, and we were given the clearance to host the meeting with minimal restrictions. So we proceeded to go for it. So we owe a great deal of thanks to Ville and the Institute of Molecular Medicine for letting us use of the facility and the auditorium. So Ville. In planning this meeting, we had to take into account our virtual effort of FlowTex 2021. FlowTex has an internet presence. There we go. As an internet presence, and we have an educational and scientific members hosted, most of which are on our YouTube channel, but we weren't just limited to Texas. These were the stats from last year with the virtual meeting. It turns out our reach also went coast to coast in the United States. But little did we know, an important caveat of the 2021 meeting is FlowTex actually reached global. We reached almost, but not all the continents. We could not leave these attendees out in planning the 2022 meeting, so we had to make this effort as a hybrid meeting, which will have to continue going forward. I don't think in the early days of FlowTex did any of us realize that FlowTex would end up having the international and educational scientific outreach that it does today. That is simply amazing, having watched from my part from about 2009 on. It's been incredible to watch FlowTex develop this way, and all because of the people and the committee involved. And it's a first-rate committee, I'll tell you that right now. In short, there are a lot of firsts for this meeting. A new venue, a hybrid meeting. We had to make adjustments to vendors who were also wanting to have an in-person meeting. The scheduling of our speakers across different time zones, including the East Coast as well as Europe. Testing the net, flipping from live to virtual presentation. Getting all possible bugs worked out of this hybrid meeting as best we can. Hopefully, even after all of this, we're hoping this meeting goes off with a hitch despite the slight delay we've had today. We hope that what you see is the effort culminating into a successful meeting with an outstanding lineup of speakers over the next 2 days in this hybrid meeting, Personally, I could not be more proud of the efforts of the FlowTex committee right now in pulling this together. With that, let's go ahead and get started. Housekeeping, badges, I think many of you know where they are at the front. Coffee and water, kolaches, waters will be coming, bottled waters will be coming. Bathrooms are around here in the foyer, as well as directly across. Lunch and vendors, you're gonna have to go through the vendors to get to the food, get to the coffee. That's the idea, is give them foot traffic. And remember, if you're doing multicolor flow cytometry, you too may be entitled to compensation. Okay, with that, it is my honor to introduce the first speaker, which should be Kathy Daniels. Kathy currently serves as the Associate Director of the Flow Cytometry Core Facility at Sana Biotechnology in Cambridge, Massachusetts. She's been working in flow cytometry for the last 9 years and is actively involved with local and international cytometry community. Her goal is to empower researchers through a better understanding of the technology while providing increased open access educational resources to cytometrists worldwide. Kathy is currently the Vice President of MetroFlow and is an ISAC emerging leader. And the title of her talk is Rigor and Reproducibility in Research Flow Cytometry from Experimental Design to Data Analysis.
Okay, thank you so much, David. Okay. Perfect. Okay. Okay, can everyone see my screen? I hope I'll take that as a yes. All right, so I'll go ahead and get started. Again, thank you so much to David and all of the members of FlowTex for putting together such an ambitious meeting. I'm so sad that I'm not able to make it in person, but I'm really excited to still be able to join virtually to talk about something that's very, very near and dear to my heart, specifically research, flow cytometry, and incorporating best practices to have our experiments be rigorous and reproducible, okay? So as we go through our talk today, we're going to be talking about what rigor and reproducibility is and why it's so important, right? We've all heard this term so many times. Why does it really matter in the research environment? Then we're going to be talking about experimental design, planning, execution, interpretation, and analysis of the data, and how we can incorporate best practices into all of those steps to ensure rigor and reproducibility. And what we're going to be doing is looking at some of the most common pitfalls that occur as we're trying to obtain the best quality results, right? So throughout my career, I've seen quite a bit of these common mistakes that are made. And the best way that we can prevent that from happening is to share our experiences and our knowledge so that other people learn, okay? So rigor and reproducibility, right? I'm hoping that my Zoom screen is not blocking anything here, my Zoom toolbar, so hopefully it's not. But we've all really heard this term before, right? Rigor and reproducibility. And we really need to truly understand what it means before we get started. So the NIH defines rigor as a robust and unbiased experimental design, execution, methodology, analysis, and interpretation of the results. And reproducibility is the ability for multiple scientists in different labs, right, to be able to reproduce those results or repeat those results by carrying out the same experiment as outlined by the original scientists. So one of the most important studies that came out in 2011 and 2012 by Bayer and Amgen was a study where they tried to take about 67 experimental studies that were reported in the literature and repeat them in their labs. Shockingly, both Bayer and Amgen found that a majority of those scientific studies were not able to be reproduced. Okay, it's important to note that a majority of these studies were in oncology research. Okay, now when they tried to repeat these studies and they weren't able to reproduce them, a lot of the original labs were saying, well, they don't have the expertise or the experience or the materials that they need to be able to repeat these experiments. And in certain cases, the original authors even went to those, or the authors of the, or the people that were trying to reproduce the studies, rather, excuse me, went to the original labs to aim to kind of rule that out and reproduce these experiments, and they were unable to. So why is this so important, right? With the importance of preclinical studies and their implication for potential studies that might make it into the clinic, we really need to make sure that the scientific community is coming together to ensure that due diligence is taken so that we can be very trustworthy of the data that we see in the literature. We need to make sure that anyone can repeat what we've done in the lab. And that's going to be really, really critical. There's a lot of money that's going into this research, a lot of time, a lot of resources. And as flow cytometrists or people utilizing the technology, we need to do as much as we possibly can to ensure the best possible results. Okay. So from there, it's important that we discuss the trends in cytometry, right? So I love showing this PubMed search, and I think many people like showing this, right? So if we look for the term flow cytometry, what we can see since the early 80s is a really sharp increase in the use of this technology as reported in the literature. So back in about 1975, that's when BD launched the first commercially available fluorescent activated cell sorter based on work by many researchers, including Len and Lee Herzenberg, right? And that led to a huge change in the field of immunology and research in general. So many, many more people were able to start utilizing this technology in their research, and that led to a really huge growth in the popularity of the technology. I like to show this example when talking about the way flow cytometry has changed over the years, right? So 9 years ago, just 9 years ago, right? I'm still relatively new to flow cytometry. What I was first trained on was the Dako Cytomation MoFo Legacy. It had this huge electronics rack, a TV monitor to monitor the stream stability, an oscilloscope, right? It took up a whole entire room. And that's what I was trained on. Okay, so it needed a dedicated operator to operate the instrument and carry out experiments for researchers. And it really enabled me to get a better, deeper understanding of the technology itself. Flash forward to today, and within the past couple of years, we now have a lot more benchtop sorters that are available, right? Like we see here with the Beckman-Coulter SRT instrument. And this is something that's more and more commonplace, right? We're getting smaller instrumentation that's being placed on benchtops in individual labs to enable researchers to carry out this technology and the experiments independently, okay? So in addition to cell sorting, when we're talking about analysis, we've seen something very similar, right? Where the technology has advanced, right? And we've seen an increase in the use of not only sorting, but also flow cytometry analysis. So what we see here first is the ICP 2022 by Wolfgang Goad from the University of Munster. And this was developed around 1968, and the predecessor was the ICP 11, and it was lamp illumination and had 2 fluorescent detectors. Okay, we move forward to about 1995 when we have the Fax Caliber, which was 2 lasers, right? And go forward to 2004, we have the LSR2, which is 5 lasers, 18 fluorescent detectors. More recently, we have full spectrum technology, which is becoming more and more popular. This is just one example with the Cytec Aurora, and it has the capability of up to 5 lasers and looking at 40 plus fluorescent parameters in our experiments, right? So in about 40 plus years, we've seen this huge increase in the capabilities of the instrumentation that we're working with. And with that comes an ever critical need to have a deep understanding of what we're doing, how to correctly carry out our experiments so that we're doing the right thing so that other people can reproduce our results that we're having on our instrument and be able to accurately analyze the data. Okay. So with that, let's say I wanted to set up a flow cytometry experiment and I asked my colleague, Rui Gardner, what are some of the best practices? Where do I start? I know that he's going to give me some best practices and some tips. So we were originally supposed to kind of chat about this together today, but I like the slide so much that I kept it in. And we're going to really now do a deep dive into some of our best practices, right? The first thing that we want to talk about today is our experimental planning so that we can ask the correct questions to design our experiment appropriately, okay? First, we need to ask ourselves, are we working with cell line or tissue? The reagents that you're going to use in order to gain a single cell suspension for flow cytometry can vary based on this. Your enzymatic or mechanical digestion will vary quite a bit across different cell line or tissue. And even if you're trying to identify specific cell types within tissue, your enzymatic digestion or mechanical might vary even there, right? So just because you're working with lung doesn't mean that you have to carry out one specific type of digestion. Maybe there's a very rare cell type that you're looking for that's a little bit more sensitive or something like that, or something that requires a bit more digestion. All things to keep in mind. And then what are the populations you're trying to identify, right? That could answer what markers or fluorescent probes that are necessary, whether or not fixation and permeabilization is necessary, if it's an intracellular marker, and it answers a whole host of different questions. So you have to ask yourself all of these important biological questions before you get started, okay? And then from there, we also need to discuss what instrumentation is available to us, right? Are you carrying out an experiment where you're going to be physically isolating and purifying populations out? Then we need to make sure we have a sorter, right? If not, an analyzer will suit the needs of your experiment, but there can be differing optical configurations across your experiment, or your instruments rather. So just because two instruments have the same name, like a BD Fortessa or even a Cytec Aurora, right? That doesn't mean that they have the same lasers, and that doesn't mean that you can pull out the same populations just because the instrument name is similar, right? And you also need to ask yourself the question of whether or not you're looking at full spectrum or conventional flow cytometry. That will change some of the, or it will not change some, but a huge portion of your experimental design and panel design as you start to consider fluorochrome choices, right? What fluorochromes are able to be looked at together, which ones are going to be pulled out by the instrument. And also, you're going to be able to start to see and think about your considerations when, or the considerations for autofluorescence, right? So if you're working with really highly autofluorescent samples, how do you approach that? The answers for conventional and full spectrum are going to be very different. And we want to take that into account in our experimental design. Okay. Next, we want to make sure we think about controls, right? So before you're at the instrument, these are all the considerations that we have to take. You need to make sure that you're going to have the appropriate controls for the experiment. At best, if you do not have the appropriate controls for your experiment, you're taking a wild guess as to where to define your positive and negative populations and how to define the biological relevance that these populations have in your experiment. At worst, it's completely detrimental and you're not going to be able to interpret any data at all. So really, depending on the controls that you do and do not include, you can have an experiment that's completely unable to be reproduced, or you can have a nice, robust, rigorous and reproducible experiment. So at least at a very minimum, you need to make sure you have an unstained control to have an understanding of the autofluorescence of the cells that you're working with, right? If you're working with more than one fluorochrome in your experiment, you need to ensure that you have single color controls. And we're going to go into all of the best practices for your single color controls for your experiment as well, a little bit later on in this talk. Okay. If you're working with multicolor flow cytometry, you also want to make sure that you have an FMO controller or fluorescence minus one. We're going to talk about that a little later. And we also need to make sure we have the appropriate biological controls, right? Like stimulated and unstimulated samples, right? Biological positive and negative controls, because we really need to make sure that our antibody is working the way we expect. And again, we're going to be talking about this in a little bit more detail shortly. Okay. One that I like to note that's very important is the use of a secondary alone control if you are working with a primary antibody that's not conjugated to a fluorochrome. The reason that's so important is because a lot of the secondaries can tend to be a little bit sticky, right? So if you're working with a sticky secondary antibody and you go ahead and you use that in your experiment, you might have some background non-specific binding. One of the suggestions I do often use with my researchers that I work with is when you're working with secondary antibody, if it's not possible for you to do an in-house conjugation or work with a vendor to get a primary conjugated antibody, at the very least, take a aliquot of that secondary, spin it down, and use the soup to go ahead and take an aliquot of that and stain for your secondary antibody, right? So that those large antibody aggregates go to the bottom as you spin it down. Okay. So what pairs in nicely with talking about controls as our next step is to talk about antibody validation. Antibody validation is one of the most critical steps in order to make sure that our antibody is actually working the way we expect, right? So in order to understand that, what we need to know is the different methods of antibody binding. You're going to have an antibody antigen specific binding via the epitope of interest, which is what we're interested in, right? We want to make sure that Fab portion of the antibody is binding to that epitope. And what we don't want to see is we don't want to see any antibody-specific binding via the Fc receptor, right? There are certain cell types that are going to express Fc receptors, and we're going to want to make sure that we carry out Fc block through a commercially available reagent or through whole serum or something like that to prevent that binding. And remember, it is a specific binding of that Fc portion of the antibody to the Fc receptor on the cells. You can also wind up with non-specific binding of the antibody, right, through excess use of the antibody, or also in certain circumstances, you might have cyanine dyes that actually bind to certain monocyte and macrophage populations, and we can get some false positives there. And you can go ahead and take that into account by using some commercially available reagents that remove that non-specific binding of the fluor to those cell types, right? But with that first method of non-specific binding I talked about through the use of too much antibody, what we can actually do is we can account for that through an antibody titration. And this is going to be really important in order to make sure that you're using the correct concentration. So the goal is to carry out an antibody titration, and typically I suggest starting out at 10 micrograms per mil and doing 8 serial dilutions down, making sure that we have a positive and negative population in the sample and titrating down. And we want to make sure that we see the point where we have the best separation of the positive and the negative populations, taking into account the spread of the negative, okay? If you don't have a negative cell that's in that population, you can always consider spiking in some negative cells that should not react with that antibody, and you could spike in your negative population, okay? I also really love this beautiful example by Kalina et al. In a very relevant volume of Cytometry Part A in 2019 that was focused on rigor and reproducibility in flow cytometry, right? So it was a great issue. I encourage everyone to check out in Wiley. And in this paper of the relevance of antibody validation for flow cytometry, they showed the importance of cross-reactivity testing, in addition to a whole lot more of information on antibody validation for flow. And here they show 2 different clones of CD85D, right, across different cell lines that were expressing different isoforms of that protein. So clone 42D1, excuse me, only showed positive signal on the expected cell line, whereas clone 287219 showed cross-reactivity and actually bound to CD85A, CD85B, as well as the CD85D. So it's something really important that you want to think about if you really need a nice specific antibody for your experiment. Okay, so use the appropriate biological positive and negative controls, titrate your antibody, and ensure that the method of binding is specific. Next, what we're going to do is we're going to talk about sample dissociation conditions. This is going to be really, really critical as you're working with different cell lines and tissue. So every cytometrist will tell you, please take a look at your cells under the microscope before you stop the dissociation. And if you were to take a look at your cells under the microscope, we're going to see something like this. And this is an example of an experiment that was carried out with a member of Memorial Sloan Kettering Cancer Center's research group and the flow cytometry core there. And you're going to see that in a lot of these slides, I have referenced some flow post-its of this collaborative work that we carried out. And they're free open access educational resources you can find on the Sloan Kettering website or the Whitehead Institute's website. But I digress. So here we can see the huge difference in different temperatures and how that takes into, how that can impact the single cell prep for this cell line. So if we look at room temp versus 4C, we see a huge increase in the single cell suspension when we go to and dissociate ourselves at 4C. In addition to that, we tried out different EDTA concentrations, and we found that 5 millimolar allowed us to have a little bit less in the way of aggregates, right? So it wasn't a huge change, but it was enough that on a larger scale, when sorting, we'd get a larger number of single cells. And we also tried out different dissociation buffers like accutase and trypsin, cell dissociation buffer, whatnot, right? And we found for this cell line that accutase was the best. Okay, so it's important to try out all these conditions and not to assume that one set of dissociation conditions will work best for all of your cell lines or all of your tissue. Okay, it's also important to make sure that with different types of dissociation buffers or fixation reagents that your antibodies will actually work, right? So trypsin, for example, can actually sometimes cause the surface epitopes to slightly alter and your antibody might not bind anymore, right? So make sure that you're testing and validating your dissociation buffers and your dissociation conditions as a whole in addition with your antibodies and your reagents. Okay. From there, as we start to talk about sample staining and washes and making sure that we have nice results, we want to talk about the number of washes that we carry out. So here in this example by Barbara and Joanna at Sloan Kettering, they showed in a very beautiful way the impact of carrying out one wash versus two washes, plural, on their staining for flow cytometry experimentation. When carrying out the one wash, as we see an increase in the antibody concentration, we see that there's not really much of an impact on the positive population, but there's a large increase in the signal of the negative, whereas that's much more mitigated as we incorporate a second wash. Okay, so when we look at this nice overlay of the data, what we can see is that the MFI of the positive and the negative, does have some slight differences, but what's really, really huge as far as the changes from between 1 and 2 washes is the RSD of the negative. Okay, so why is that important? When we take a look at our stain index, we can see a huge difference between the 2 washes and the 1 wash in terms of separation, right? So that's going to be really, really important. What you want to keep in mind, though, is that as you increase the number of washes, you can actually start losing some cells in terms of numbers. So you want to make sure that you factor that in as well. So you really want to evaluate specific to your experiment and your staining workflow to make sure you're getting the optimal signal while also keeping the cells that you need for your experiment. Okay. The next thing we're going to discuss as part of our staining workflow is the importance of taking into account the fluorochromes that you're working with. So in this example, we have BV605 and BV711, and these are 2 polymer dyes, right? Polymer dyes can actually have an interaction with each other, and that results in data that looks like this, where we see a pattern of undercompensation, right? So because those polymer dyes are interacting with each other, we can see a bit of an issue, right? So when I first saw this data set, I saw that indicative pattern of undercompensation. So in order to kind of put my investigative hat on, I decided to look at the compensation controls in the spillover matrix to make sure it was correct. And I see that the compensation, aesthetically looking at it, was spot on, right? So just a visual inspection, right? Spot on. And then what I realized was that when changing to a staining buffer, like Brilliant Stain Buffer, we take into account that polymer dye interaction, and we now have a beautiful representation of our data, right? And I'm saying but this is actually wonderful work that was done by Anna-Laure Iscache at Memorial Sloan Kettering, and she's kind enough to share the data for one of these flow post-its. So, we use this as an educational example of why the Brilliant Stain Buffer is going to be really, really important. And it's also available from other vendors as well. Okay. The next thing that we're going to talk about is autofluorescence. So here in this example, what we see is a highly autofluorescent parental line that's in blue, overlaid with a sample that we expected to have M. cherry in red, right? And by looking at this data in a univariate fashion, what we see is we're not really able to parse out which cells are M. cherry positive and which cells are M. cherry negative. So one of the solutions here is to look at your data accordingly and display your data accordingly. Avoid those histograms and plot your fluorescent parameters of interest versus an open channel used to detect autofluorescence. So this is that same parental sample. And this parental sample, we're plotting it M. cherry versus the 515 to 545 nanometer filter that's off the blue laser, which is a typical detector where we see high levels of autofluorescence. So that when we go ahead and look at it, we can identify where that highly autofluorescent population is. And when we look at our experimental sample, we can now clearly define where our M. cherry positive is, pulling it out from the autofluorescent sample, okay? Other things that we could also do is we can go ahead and we can choose fluorochromes that are not in the typical excitation and emission range of the autofluorescent molecules in cells. Or we can consider using full spectrum flow cytometry technology, which enables us to reduce the impact of autofluorescence, which is what we're going to talk about next. Okay, so this is an example of a data set, and using conventional flow cytometry techniques on a 5-laser Cytec Aurora, we just plotted our unstained, which is an orange, versus our fully stained sample, which had BV421 and Alexafluor 488. Just looking at a V1 detector and a B2 detector, which is similar to what we do in conventional cytometry. And we can't pull out what's truly double positive because of that overlay of the unstained and the highly autofluorescent population. So if we then look at full spectrum flow cytometry and we carry out our unmixing, we see it still doesn't look that great, right? We see this kind of spread of the negative, and our positive doesn't pull out all that well, because we're just seeing this weird pattern. But what we didn't do just yet is we didn't take into account the autofluorescence and treat it as if it's its own spectral signature. By doing this on the Aurora and on other full spectrum units, we can actually treat that autofluorescence as if it's its own separate parameter and better separate the unstained and the fully stained samples. So you can imagine it would be really hard for anyone to interpret this data in a conventional or in a full spectrum manner without taking into account the autofluorescence. But once we do, that's a huge change to the data, and it's a great improvement that enables us to accurately read out the results. And if we look at an overlay of those three different signatures of BV421, Alexafluor 488 rather, and our cellular autofluorescence, we can see why this is so important. So looking at the whole full spectral signature of our cellular autofluorescence, it's so high that our Alexafluor 48 just pulls out and is a tiny bit brighter on our primary detector there, similar to our BV421. Okay. So that's why it's so important to understand the autofluorescence of your samples before moving forward with your experimental design. Okay. Next, we're going to talk about, a different aspect of autofluorescence and how to set gates, right? So Oftentimes, people will just look at their negative control to set their gates, but that's not always enough, right? So sometimes their unstained population, just looking at autofluorescence is not appropriate. And that's where we want to look at fluorescence -1 controls or FMO controls. So this great example by Stephen Perfetto et al. Back in 2004 shows us what we can accomplish by utilizing FMO controls in our experiments. By adding in every single fluorochrome with the exception of one, we can see the spread of the data and be able to more accurately set gates. So in this experiment where they have FITC, PE Sci5 and PE Sci7, if we use the unstained gate to define PE positivity, it would not be accurate. And we can see this here with our FMO control, because there's spread of the FITC detector into PE. It's really important to note that this is not due to compensation. This is due to photon counting error, and it's just revealed due to compensation, right? So having a compensation of 50 versus 70 is not going to mean that you have higher spread or lower spread, and we're going to talk about that a little bit later. Okay. But this is a great control for determining positivity. And it's also important to note that we don't need to go ahead and just use one FMO control for every single detector or reagent in your experiment, rather, but use it for the most important markers, the ones that you see a continuum of expression or difficulty determining positivity. Okay, next we're just going to very briefly talk about dead cell removal and its importance in the rigorous design of experiments. So you can see without a dead cell exclusion, if we're looking at CD4 APC versus side scatter, we have about 20% positivity for CD4. But with dead cell exclusion, we're able to see that same population is now only 10.9%. So this is the same exact sample, just with and without viability dye. And that has a huge impact on the statistical analysis of your experiment. So you always need to make sure that you're carrying out a dead cell removal. And you want to make sure that you're actually titrating those dead cell removal dyes, right? So if it's an arc-amine dye like we see here, an amine reactive dye, I should say, we see that the unstained population is right here. And if we're using the dye at 1 to 1000, we see a shift up in the negative because it's staining the surface amines. So what we'd like to do is titrate your amine reactive dyes for fixable or live flow cytometry experiments down, so you get a separation of the positive and negative without seeing an increase of the live cell population due to amine staining. Okay. And you also want to take this into account with DNA binding dyes such as DAPI or PI, right? Carry out a titration to make sure you're using the correct concentration. So we always want to make sure we're incorporating dead cell removals into our experiments. In addition to that, we need to make sure we're taking into account when you have a lot of debris, right? So if you're working with tissue or sample that you expect a lot of debris, you might have a DNA binding dye that you're using in order to determine, which cells are dead, but your debris won't stain positive for those DNA binding dyes. So you can have a really hard time determining what's debris and what are our live cells. Utilize a live cell marker such as calcine, right, that gets into the cells, and if they're cleaved due to esterase activity, if the cells are metabolically active, you'll see that positive signal and be able to determine much more accurately where your live cells are, so that you're not getting the detrimental impact of the antibodies non-specifically binding to dead cells and debris, okay? And this is that same data set, and it shows the importance here in red and black as the overlay of the calcine AM negative and positive, as the importance of not using scatter for a viability determination. You cannot set a scatter gate and just say, oh, these are my live cells, it's fine, right? You need to make sure that you're taking into account a viability or a vital marker in your analysis, or else you can have skewed results that are not repeatable. All right, I'm actually going to skip over this slide real quick, but it's important, because we're on a little bit of a time crunch today to talk about instrument setup in QC. So this is just a quick data set to show the incorrect laser delay and the correct laser delay on the same exact sample. And this is actually a sorting experiment that I had carried out with a colleague at the Whitehead. And we realized that the laser delay was incorrect, right? because they noticed the population didn't look as expected. So it's a huge difference in the double positive population, almost 8%, right? So you always need to make sure you carry out the appropriate instrument QC so that you're laser to laser accurate, because it will result in incorrect data if the laser delays are not appropriate. And we also, when we're actually at our instrument and setting up, what we want to make sure that we take into account is the impact of different instrument settings on our experiment and the results that we see, okay? So everyone will always tell you what's best to do is to concentrate your sample and run them at a low flow rate. Why is that important? We all know that the sheath pressure remains exactly the same throughout our run if we have a nice stable fluidic setup. And your core stream is widening as you go from low to medium to high. And what can happen there is you can increase your chances of coincidence events and have a higher electronic abort rate, which we don't want. But as far as the data itself goes, if you start to widen that core stream, you want to just be very mindful that your cells might be then hitting the edges of the laser, potentially. And because of the Gaussian distribution of power of the laser, what can wind up happening as you go from low to medium to high is you can see an increased spread of the data. And this is from Rui, so thank you very much for this data set. And we can see that as opposed to this nice tighter CV that we saw originally with the sample on low, when we went to high, we saw a spread of the data and a decreased overall MFI. So if you do have to change from low to medium to high between samples, do a QC check to make sure that signal is not drifting. Because if you're making those manipulations and changes to the instrument, what can wind up happening is you can get different results from a statistical perspective, right? When we're looking at the spread and also the MFI, and you can think that's biologically relevant, right? So make sure that you're checking that in your run. Okay, so yes and no. Okay, one of the next things that we're going to be chatting about today is off-scale data. Okay, this is irrelevant to what fluorochromes there are, so I didn't put any axes on here. But it's one of the things that I've seen quite often in the literature, in data that's presented to me, and it's something that I try and talk to people about quite often, right? So if we have data that's off scale, what it's essentially like is taking a picture of someone with their head cut off, and that's the analogy that I always use, right? So in this example, we see Marge Simpson, but we see her in Simpson, and we see her in a much different way than we usually do, right? Her iconic hair is completely gone. So if someone were to look at this without having known this character, they'd say, oh, completely normal, that's fine, right? But we're missing a huge aspect of this cartoon. The same thing goes with your flow cytometry data. Populations can be hidden if the data is off scale. The MFIs will not be correct if it's beyond the linear range of the fluorescent detectors. And it can result in incorrect compensation. So for all of these reasons, your data will not be appropriate for reporting, and you need to make sure that this does not occur, right? What I typically suggest before you start your experimental run is, or recording all of your samples, I should say, and even before starting your comp, run a little bit of your fully stained sample that you expect to have the brightest signal in all of your detectors, and make sure all of the signals on scale before proceeding. Again, we always want to make sure, rather, that your signal and all of your samples are run at the same voltages or gains from sample to sample, okay? Next, we're going to talk about PMT balancing, our next aspect when we're talking about how to set voltages, right? So this is a really, really great example on trying to reduce compensation by adjusting voltages. So we call this PMT balancing. A lot of people do it. Is it really necessary, right? So in this example, we see that our APC size 7 is at a fixed voltage of 580 volts, and our APC is at first at 400, and the compensation is 110%. If we go up to 415, that compensation lowers to 82.8. and so on and so forth until we get a low compensation of 40.5%. Has it improved the data at all, though? We should ask ourselves this. So even though we had a compensation of 110.2, Our stain index for the separation of APC positive and negative is 45.7 at the highest comp, and it's actually at 40.7 at the lowest comp with this voltage right here. So we reduce that compensation, but we're not able to see the separation as well, right? The other thing that's really, really important here to note in this beautiful example is that the RSD of the data, or the standard deviation of the data, remains largely the same. So you're not seeing a decreased spread as you lower that compensation. Harking back to my earlier point that it's the spread that we see is due to photon counting error, and compensation shows us that that's occurring in our experiments, but it does not cause it, right? So you don't have to really worry about sitting there and kind of adjusting voltages and going crazy to keep everything below 100. Make sure that you get the best separation with your fully stained sample and with your single stain cellular controls to ensure you're getting the optimal stain index. And don't worry too much about trying to reduce those compensation values. Okay. Okay. From there, we're going to have some comments on compensation and unmixing and the best practices to ensure that you're able to reproduce the data across sites, right? So we always have this drilled into our heads that we need to make sure our single color controls are as bright or brighter than our experimental samples. So this shows us in this data set an example where we see a clear overcompensation in our experimental sample in red. So I overlaid that sample with our single color control for PE size 7, right? So this is between PE size 7 and PE. And what I see is for my single color, everything is compensated perfectly. But once our sample is brighter for that specific fluorochrome, overcompensation. So it's really, really important. And what I tell people is whenever you see extreme negative populations going into the -3rd, -4th, there's usually some type of an issue that we're seeing, and you want to investigate and put your detective hat on to troubleshoot your data. Okay. It's also important that we realize that when we're setting up our compensation or unmixing, you take into account the spread of the data, right? So for this example of Alexa 647 and DAPI, if we set the gate on this bright portion of our Alexa 647 positive and we carry out the comp, we see that it's overcomped still. And our MFI of the positive for the single color control was 26,050. If we adjust that positive gate when carrying out compensation, just take the really brightest portion of the positive, our MFI is 139,203, and it's taking the brightest portion of that to correct for that spectral overlap. And now our populations look much better as far as compensation goes. So make sure you have a nice bright sample for your comp or unmixing. Okay. The next thing we really want to drive home is the importance of compensation controls, right? And making sure that our fluorochromes match. So in this example, if I look at Alexa 700 and APCR 700, what we see is the emission spectra is almost identical. If I look at the 730-45 filter, that's the filter that we're using to detect the light that's given off the red laser, which both of these fluorochromes are excited by. Okay. If I try swapping out one fluorochrome for the other in my experiment, it's not going to work, right? So even though I see that they're almost identical, if I try taking APC R700 and using that as my single color control for compensation or unmixing in combination with APC, but then in my actual experimental sample, they're dual stained for APC and Alexa 700 here in blue, huge issue. So the instrument itself and the software and the algorithm that's in place to compensate or unmix is doing everything correctly based on what it's given. So if I look at the APC R700 single color, it's compensated or unmixed perfectly. But because we had a different fluorochrome in our actual experiment, we're seeing that pattern of overcompensation. Okay. So the single color controls need to have matched fluorochromes to your experiment. Next, we're going to talk about the importance of using the correct autofluorescence, because this is one of the most common things we see as far as having incorrect compensation or unmixing results. Okay. So if we look at an overlay of unstained cells in blue and unstained beads in orange, we see a clear difference in autofluorescence off the FITC detector. if I look at that in a dot plot versus APC size 7, we can see the same, same data just plotted in a slightly different way. And if we try to compensate using the incorrect negative autofluorescent population, we see that data here in this second data overlay in red. So if I say everything just has the negative of beads, the algorithm is going to calculate it appropriately based on the input of what we've told the instrument and the software itself. So it's going to accurately correct for that spectral overlap, taking into account the negative population here that we see. But if we tell it the correct population, that compensates absolutely correctly. So, the instruments will do only what we tell them to do, and we need to make sure that we have a deep understanding of the technology itself so that we're getting the best results, right? So make sure that when you're compensating or unmixing with different material, you reference the correct negative. Okay. And this is just a very quick data set to show, that not all cell types will have the same autofluorescence. So this is a beautiful example from an updated guide to the perplexed for cytometry in the high dimensional era that was published in 2021. And it shows the plot of CD11C versus CD19. And the differences that can occur in autofluorescence between different cell types, right? So you can see here for our CD3 positive T cells and our CD11C positive conventional dendritic cells, we see that there's what looks like perfect compensation, right? And some people might look at these CD14 positive monocytes and think that something's off as far as the compensation or unmixing goes. But that's really just a difference in autofluorescence between these populations, and it shows it very clearly in this data set, okay? And you want to take that into account in your analysis. Okay, so now this one is very, very dear to my heart, and it's something I've been talking about with people quite often, and one of the most important slides, personally, I think, in this example, or in this talk, rather. So I looked at this data set, and it was marker X and marker Y. I'm not going to say the fluorochromes, right, because it's not really of interest to me at this point. And I saw what looked like an undercompensation. I said, the algorithm didn't work. Something happened. I don't trust Diva. I don't trust FlowJo. I don't trust, Site Expert, whatever software I'm using. The compensation kind of stinks, right? So I decided I wanted to compensate or unmix and tweak it by eye. So I changed the compensation from about 0.2964% to 3%. And I see my data looks gorgeous, right? I have this nice pullout of our populations, and it's compensated or unmixed beautifully. So I'm a genius. I did everything perfectly right. Then I was curious, right? I'm always curious. I want to take a look at my data and see what caused it. So I overlaid my single color controls with my experimental sample, looking at that new comp matrix or unmixing matrix. And what I see is, wow, my single color control no longer correctly compensated. Now it's overcomped. So I tried to identify what happened there, right? And I looked at another sample in my experiment, and now that shows overcompensation with the compensation that actually was something I just corrected for another sample. So I looked at an overlay of those two samples to see if maybe there's a different intensity of the signal. Exactly the same, right? This was just a duplicate sample. So why is one showing overcompensation, but I fixed the other one and it was fine? The reason for that is because it was actually a polymer dye interaction. It's that original sample I showed you. And the reason why we need to use those brilliant stain buffers when we're having two polymer dyes interacting with each other. So what I did was I went ahead and said, I know better. I'm going to compensate this by eye and fix my data. But what I really did was I masked something that was actually happening between two polymer dyes in my experiment. So how can I say that I can tweak this data and it's repeatable to some other lab across the pond or somewhere else, right? So over in Texas, if someone else was trying to repeat this, but I just went boop, boop, and I made a couple of changes to the comp matrices, how would they know how to do that? And is that rigorous and is that reproducible? Because this was really a problem with the experimental design. Okay, so how do we know better if we don't know everything about our experiments, right? We can't just manually tweak compensation and unmixing and report that data in the literature, because that is not reproducible. Okay. And that's something that's very near and dear to me. I might have some people shaking their heads, but it's something that I feel very passionate about, and something that I think we need to address as a community. Okay. In addition to that, it's important that we make sure that we're running all those appropriate biological positive and negative controls for our experiments, and also making sure that we're running our appropriate single color controls for all of our experiments. Because if we try to recycle old spillover matrices, we're going to see something like this. In the red is an overlay of our recycled compensation. In green is our same day compensation with fresh controls. And that can have a very clear impact on the data. So if you don't take into account the differences in the instrument performance and the reagent changes from day to day due to potential tandem dye degradation or exposure to light, or any differences there, you're going to have incorrect data. And we already know that we shouldn't just be manually tweaking data for compensation or unmixing, because that's very incredibly subjective and not reproducible. Okay, so you want to make sure that you're running your single color controls each and every day. No matter what the vendor tells you, you don't know and you can't capture any changes in the reagents themselves by recycling old spillover matrices. Okay, so the instrument might be stable as anything. But what about your reagents? Okay, so just don't do it. Okay. And the last thing we're going to be talking about before I end for today, and hopefully I'm on time, is the differences in data scaling, right? So when you're reporting your data, what you want to make sure that you do is report it appropriately. So here I see 3 different examples of the same exact data set displayed in 3 different ways. We have log, which doesn't show us the full display of data around 0. Then we have bioexponential, which is showing us the linear log display around 0, but it's not optimized, and we see an excessive amount of white space around 0. By optimizing that bioexponential display, whether it's in the acquisition software or in our data analysis software, we're able to get the best resolution of our populations, and that's what we should aim for. Okay. So make sure you scale your data appropriately to best visualize your experimental data. Okay. From there, we can wrap up. And this is just an overview of what we chatted about today. So I want everyone that's there and listening in today to ask yourself all of these questions. Is your experimental design and execution rigorous? Was your dissociation optimized for your cells of interest? And are the results consistent? And are those reagents appropriate that you're using to detect and accurately pull out the populations using the antibodies that you're using? Is your sample staging in accordance with best practices through the use of a viability dye, the correct buffers, and the appropriate reagents? Were your antibodies validated? The instrument set up and used appropriately? And are your populations clearly defined and interpretable? Can this data be reproduced, right? Were the controls appropriate to allow for the correct interpretation of the results? And can it be transferred to another lab without saying, you might have to tweak this or that, or kind of play around with this? Or can you just give them an SOP and it works for them as well? Is the data displayed appropriately? And all experimental conditions clearly explained? And lastly, and most importantly, did you have to manipulate the results in any way to get expected staging patterns? If so, that data is not reproducible and very subjective. Okay. So with that, I think I've hit my time, and I just want to say thank you so much to the members of FlowTex for inviting me here today. And though I'm not working there anymore, the colleagues that I had at Memorial Sloan Kettering were absolutely instrumental to today's talk, and I can't thank them enough. And I didn't get to work with her myself, but I had to add in Anne-Laure Iscache. She joined after I left that lab, but she has been absolutely amazing to collaborate with on some flow post-its while I was still taking part in them. So Anna, Gus, Mark, Fung, Joanna, Rui, Barbara, Ben, Fabio, and Magda, thank you so much, and I miss working with all of you. And with that, I think we might have a minute or two for questions, and we're all set.
Okay, fantastic talk, Kathy. We do have a couple questions from YouTube, and if anybody has questions here in person, you can, there we go.
You use the mic because Kathy won't be able to hear you.
Actually, don't know if that's on yet.
Perfect. I like this interesting Q&A session, virtual, hybrid, in person. It's great.
Hi, Cassie. That's nice talk and very useful information. And I have one question about that off scales you show. That's good examples. But I saw that lactive and positive have a so big difference. But my question is, what do you think if that is the case? What do you suggest the LAX experiments to optimize these color stainings?
That's a really great point, and thank you for such a wonderful question. So what I would suggest in this case, try two things, right? So maybe one of the things that we see here, if you can see, is there's a pretty big spread here in the negative. So there's a high chance that this antibody can actually be titrated down. We might be using such an excessive amount of antibody that we're overdoing it, right? And then we're seeing that big increase in the negative population. So I would first suggest to titrate that antibody down and make sure that that antibody has been validated and titrated. And then from there, I would always suggest to make sure that you put on an aliquot of your fully stained and really adjust it, right? Your negative population being a little bit lower is okay, as long as your positive population is on scale. There also is the option of swapping out that fluorochrome, right? Maybe, for example, this is a fluorochrome that's screaming bright, and it's not necessary for this marker. So if it's a marker that there's a huge number of receptors on the cell surface, if it's a cell surface marker, and you're pairing it with a really bright fluorochrome, you can pair it with something dimmer so that you're not seeing that crazy high signal if it's not necessary. So those would be my three suggestions. Bring your voltage down, titrate your antibody, and make sure it's used the appropriate concentration, or switch the fluorochrome.
Hi, Kathy. Thank you. Everything was wonderful. You may have just answered my question. If you remember the slide where it was an unmixing control, where you adjusted the positive gate to very bright, it seemed to me that that data was also off scale. So was that just an attempt to salvage that experiment, but that positive control being way on the right is bothering me.
Yeah, so that's a great point. So usually what I suggest doing, this is very close to being off scale, so there's a chance that this voltage could have been brought down just a hair, right? And I agree with you on that. If we brought that, this is an example of data from the BD instrument, and usually those detectors are linear to about 200,000 or so, which is that first tick after 10 to the 5th. And here, if we brought that gate even further up, we'd probably see much less than 1% that was actually truly off scale, right? So in an example like that, this voltage might be okay, or it brought down just a hair. We brought that gate up higher so that we had a more accurate compensation. But at the end of the day, if we just set a gate on what's truly off scale, which for BD instrumentation, I typically tend to say the first tick after 10 to the 5th is where we don't want any of our data to be due to the linearity of the detector. If that's below 1%, I treat that as okay, like 1% or 0.5% should be okay. And if we'd adjusted that gate, likely it would be. It's just because we set it at about 1.34 so that we had enough events, statistically speaking, so that we could actually carry out the compensation algorithm. It's a great question.
Four questions from the net, but I think because of time, I'm only going to be able to grab one of them.
Okay.
What is the optimal ratio of brilliant stain buffer to perm wash buffer if you use multiple polymer dyes for intracellular staining?
So if you're using multiple, as far as the fixed perm question, I can't answer that specifically. I'd suggest reaching out to maybe your rep from the company that you're working with for an answer there. I will say that a lot of people don't realize that there are actually concentrated brilliant or super bright stain buffers that you can use that minimize that minimize the volume that you have to add. So if you have a 20 color experiment, or sorry, 20 floor experiment or something like that, you can actually work with the concentrated buffers to counter that polymer dye interaction. And I would suggest trying to work with those that you're not using such a large amount of the brilliant stain buffer. And that might be helpful. But for the fixed perm question specifically, I'm not super educated and I don't want to steer you in the wrong direction.
All right, thanks. For those questions I didn't get to, we'll make efforts to answer the questions offline. We're going to move on to the next speaker.
Thank you so much, everybody.
Good morning. It is my great pleasure this morning to welcome our next speaker, Sarah Schnieder. Sarah obtained her bachelor's degree from Clark University up in Worcester, Massachusetts, and then came back to Houston, where she is natively from, and worked for a while as a research associate at Texas Heart Institute, and then came to my lab, the Advanced Cytometry and Cell Sorting Lab at MD Anderson, and stayed with us for 5 years. The work she'll present today is part of the work that she did as a collaboration while she was with the ICSF. And Sarah is now a graduate student candidate, a PhD candidate at the University of Texas GSBS. So please welcome Sarah for her talk, Probiotics and Immunotherapy in Melanoma and Computational Workflows. Thank you, Sarah.
We share presentations here. Almost. All right. Since we're running a little bit behind, I'm kind of going to fly through this, but luckily Kathy already covered a lot of the stuff that I needed to, so that's good. Like Karen said, I'm talking about a collaboration that we had with a group at MD Anderson who looked at probiotics and immunotherapy in melanoma. And I'm going to talk a little bit about the data, but I'm also really going to focus on how to do this computational analysis workflow for flow cytometry. So the 3 aims, we're going to talk a little bit about what computational analysis is and why we use it in flow. Then we'll do an overview of the paper, and then I'm going to give us some point-by-point steps for at least how I did this particular analysis and some best practices as well. Okay, so what is computational analysis? Basically, it's mathematical modeling tools to visualize and interpret complex data. So there's 2 major components that we talk about with computational analysis that we use a lot in flow. The first one is dimension reduction, which is a tool for visualizing the data. And basically, we would take something you can have, maybe spectral flow that's 30 parameters or 40 parameters, and we're going to drop it into just these 2. And so some examples of that people are probably familiar with is Tyznik, UMAP, and principal component analysis is one of like the original dimension reduction tools. And then on top of that, we're going to include also clustering. So what clustering does is it creates groups, or we call them clusters, of the data based on similarities. So in the case of flow cytometry, that would mean that we have unique cell populations that should be together in one cluster. And so some examples of that are FlowSum, phenograph X-shift, and k-means clustering is another one. So just what you can see on the plots here, this first top one is just an example of dimension reduction. So we've taken, in this case, maybe 12 different parameters and squished them into 2, Tyznik 1 and Tyznik 2 on the X and Y. And then on the bottom, we're still using that dimension reduction, but now we've overlaid the different clusters on top of it. And so you can see that the algorithm on its own has picked out what the dimension reduction did, but really it's picking out these different phenotypes as well. And so we'll get into that a little bit more. Okay, so why do we use computational analysis for our flow cytometry data? Like I mentioned, it helps get complex data all together so that it's easier to interpret. So it's good for high parameter data, like we talked about the spectral cytometry. We have traditional cytometers that have high parameters, and even if you're just using, I guess it's kind of common, at least at the cores, it's commonplace now to have like a 10, 12, 14, 15 color panel. But it's not always easy necessarily to analyze that. It's also good even if you don't have these super high parameter, if you have a large cohort. So if you have, you know, maybe 50 mice that you've looked at over a year, it's going to be a little cumbersome to put each of those cytometry plots together over and over and over. And same thing if you have multiple time points, different treatments, different genotypes, that kind of thing. So we kind of get to take our traditional cytometry analysis, which we can see on the left, top left up there, and we can also consider all of the parameters at once, you see in the top right, and we put them all into one plot that you can interpret. One of the benefits, or the benefits of it to me, is that it's unbiased confirmation of these populations that we've been kind of picking out on our own. And it also helps us pick out these differences between groups very quickly. And then it does also have this kind of discovery aspect too, which is that if you're not looking at one marker on a certain population in your traditional analysis, you may it may come up in a cluster, or it may come up in the computational analysis too. Okay, so we're going to talk about this paper. It just came out from at the end of last year from Jen Wargo's lab here at MD Anderson. The title of the paper is Dietary Fiber and Probiotics Influence the Gut Microbiome and Melanoma Immunotherapy Response. And I know this is a little bit of a dizzying list of authors, but the reason I put it up there is to emphasize that a lot of people worked on it and put in a ton of effort on that. And my piece in this was really just particularly this flow cytometry analysis, but these people really all deserve much more credit than I do. Okay, so here's a quick overview of the study. They had a huge cohort of melanoma patients, over 400, and these are people who, for the most part, were receiving immune checkpoint blockade therapy, anti-PD-1 therapy. And they looked at a few things. The first they looked at was at the gut microbiome, and to see if there was any kind of differences, or if there was any trend in the responders and non-responders. And then the second thing they looked at was these dietary habits and the use of probiotics. The reason being that many people, I should say, that are going through this immune checkpoint blockade are also taking probiotics, you know, with or without any necessarily indicator of that. After that, what they did is they created some preclinical models. So they gave these mice FMT, a fecal transplant, and then they treated with probiotics or without, and they also treated with anti-PD-1 blockade. So they compared with and without probiotic, and then they also compared a high and low fiber diet. So the first thing that they realized is that the probiotics actually reduce tumor control in the microbiome. Oh, I wish, hang on. Hopefully that goes away. Okay, anyway. It's kind of blocking it out, but it reduces tumor control and microbiome diversity in these immune checkpoint blockade treated mice. So what you can see in A basically is that the mice that were treated with immune checkpoint blockade and also were given probiotic have lower survival. And then we see bigger tumor size in C, and then in D we also see, this is looking at complexity in the gut microbiome, and we see a decrease there too. We see the same thing in E. So just kind of the first piece. Okay, the second piece is that they looked at the immune system, these tumor infiltrating cells, and in particular, they saw that cytotoxic T cells, especially the ones that are interferon gamma positive, inflammatory, are also reduced when taking probiotics. So same thing, they're treated with anti-PD-1 antibody, they're given probiotics, and we see this pretty significant reduction in F for both types of probiotic, and there is kind of a trend, but not a significant reduction in the interferon gamma CD4 T cells as well. And just at the bottom in H is what I'm going to talk about and how we actually got that data, which is computationally analyzed. But you can see all of the different immune subsets that we picked out from not a terribly complex panel, but it still became interesting data. So this is in line with previous studies, what they found, and basically it requires more studies. And I think they talked about optimizing probiotic formula. The other half of this paper was that they tested out the high fiber diet, and they did see that higher fiber improves tumor control in these immune checkpoint blockade treated mice. So we'll see in the top, in A, that if they had sufficient fiber, which is the high fiber or normal fiber, they had increased survival. And same thing, they saw with high fiber and no probiotics had increased survival or the best survival. They saw additionally reduced tumor size in D in that green filled in dot with the high fiber and immune checkpoint blockade. And they saw distinct microbiome in E. The last thing they did was some RNA-seq, and they found that these tumor infiltrating T cells are activated basically. So they have, you can see in G, the highest upregulated genes are T cell receptor signaling, T cell activation, interferon gamma, basically a whole bunch of immune stuff. Okay, so a little overview of the paper, and we'll talk about the computational analysis that I did for that. So first, it's just this data is a 10-color T cell panel, and it's kind of just looking at the some of the major T cell subsets. And what we did is took tumor from 20 different mice, and they were in 3 groups. So a control that was just given water, and then 2 different probiotics. We had a few different groups. So luckily, Kathy really already covered this for me, so I won't talk about it much. But the first thing you have to do before you run your data through the computational algorithms is to clean it up. So get rid of debris, get rid of the aggregates, get rid of the dead cells. A couple other things that you really ought to look for is this inconsistent flow rate. It pops up all the time, or with some regularity, at least. So you can either, you can gate it by yourself, or you can use these other algorithms that are in FlowJo and I think a few other programs, FlowAI and FlowClean. And then, like she mentioned, there are these antibody aggregates that can form with the Brilliant dye. So use that Brilliant stain buffer if you have it. The next thing that you're going to do once you have your individual files cleaned up is that you need to concatenate them, which means bring them together. So before you take all of your files that have this distinct information and squish them into one, you have to make sure that you can deconvolute that later. So the way to do it is by adding these keywords. And when you add keywords, so for example, in this data set, I had experiment, tissue type, and treatment. And you need to make sure that they're numeric integers so that you can pull them out, because when you add the keyword, it will become a parameter that goes onto a flow plot. So you have to have a number to pick it out. It's a good idea between your groups to set kind of an even number of events. It might you just have some differences. So what you can do is you can either use the downsampling algorithm, or you can set a maximum number of events per file. And then when you're concatenating, also make sure that you include those keywords in that additional parameters kind of at the bottom of that screen. By the way, if it's not clear, I did this analysis in FlowJo, but it can be done in other software as well. Okay, so the next step, you've concatenated your file. You now have just one file, but you have to do a few things to it first. The first thing is that you should gate on your groups that you created. So if you look kind of in that middle figure, you can see now what was before 20 mice is now just 3 single lines, and those represent the different treatment groups. And so you can draw a gate on them so that you can get back to that later. The other thing you should do is adjust your parameter scaling, and Kathy talked about that. It's good for visualization. It's required for these algorithms. What you see is what the algorithm sees. So if it's all squished up on one side, it's going to think that the information is squished up. So just make sure that you spread out your positive and negative on the scale. And then you can run your clustering algorithms, and those are usually downloaded as plugins. One kind of caveat, a good rule of thumb, unless there's a good reason for it, you should exclude the parameters that have already been gated out for manually, like I gated out the live dead, so I don't need to include that anymore. And you can, there's a few different strategies, but this is how I did this data set in particular. So now you have your dimension reduction map, and then you have some clusters as well. So on the left, this is just the dimension reduction on its own. And we see we've got kind of 4 big lobes and a bunch of other ones, but they don't mean very much by themselves. One good practice is to overlay these groups. In most cases, they should intermix pretty well. And if they don't, you might be experiencing like some batch effect, which is when you run them separately. The machine is a little bit different every time. We should run those quality controls on the machine, but anyway, it's a good idea to make sure that they kind of go into the same place, because what we want is for those pieces on the map to represent a different cell type and not a different mouse necessarily. And then finally, we've just overlaid onto that those flow sum clusters, just like we kind of showed in the beginning. Okay, so now you have a pretty picture with lots of colors and dots, which is our favorite thing in flow cytometry, but you have to figure out what they actually mean. So there's a few good ways to do it that I like. You can look at these unbiased clusters, the flow sum clusters, and compare them to manually gated populations. We should all probably have a gating strategy. So for example, now I can see that what's orange in the flow sum, cluster number 3, corresponds to B cells. So I can probably identify that. And then if I look down at this overlay map in the bottom left, I can see, okay, CD19 is my B cell marker. Those are probably B cells. And then if I go to this heat map that FlowSum generates as well, I can look for cluster 3, and I wish I had it, but it's got CD19 in it. Okay, so we did that, and this is the exact same thing that happened here. There's a few other things you can do to pick out the pieces, and what we did is we overlaid based on these groups that we had separated later. And now we can see that some of the clusters that are falling in the same place are there in one type of group and not there in the other. And I'll point out kind of in the top left, this purplish, and then the bottom left, that purplish. Those are the cytotoxic T cells, which we saw decreased, and they're basically gone in that probiotic 1. And then once we took the statistics, they were pretty much gone in the probiotic 2 as well. So this is just a nice way to take all of that data and put it together. The last thing that you can do if you want to get your frequencies, which you probably do, you can copy those flow sum clusters, which come up as gates in the workspace, onto the different groups that you had separated initially. And then these computationally derived population can also be applied back to the sample ID that originally came with it. So you can say like mouse number 1 has 5% of cluster 8 or whatever. And you can run that through like t-test and all kind of normal statistical analysis. And then finally, if you have a more complex data set, you could isolate, for example, those cytotoxic T cells. And if you had a bunch of cytokines or transcription factors in the panel, you could repeat clustering just on that one piece that was there. Okay, so just a review of what we did as fast as I possibly could. And here's those steps again. So it's just reminding you, you can get a lot out of it, but it's super important that you clean up the data. You have to concatenate the file. When you run the algorithms, you also have to kind of pre-work that file, and then you can start to get your maps out, pick out what your clusters are, and export your statistics. Big thanks to the Wargo lab. There's a whole bunch more people in there, like you saw, but Alex Cogdill is the one who was working with me on this, and then the Wadowich lab did the flow cytometry. And of course, the core, Karen mentioned, is where I was when we were there, and she helped me out a lot on this too, along with everybody else. And that's it, thank you. I like FlowSum because Oh yeah, sorry, repeat the question. He asked, why did I choose FlowSum and not another clustering algorithm? I like FlowSum because it gives that heat map, so I like it for the cluster identification. Also, I think with these lower parameter panels, when you do put them in, sometimes something like Phenograph ends up giving you a bunch of clusters that are artifacts and not real. So I like in FlowSum, it's like unbiased, but you give it a little bit of bias by telling it how many meta clusters you want. Okay, sweet. Thank you. Oh yeah, we do have a short break, although we've kind of gone through like half of it already, but let's do maybe 5 minutes and then we'll come back. Yeah, 10:30. At 10:30, we'll come back. Okay. We are ready to go again. All right, so our next speaker is our very first emerging scientist, although I'd say she's pretty well emerged at this point. So next speaker is Sofia Mastoraki, and in 2018, she received her PhD from the University of Athens, Greece. Her PhD thesis work focused on the discovery of the clinical evolution of blood-based DNA methylation biomarkers in liquid biopsy samples of breast and prostate cancer patients. She's currently a Triumph postdoctoral fellow in the QMRC Hunt lab at MD Anderson Cancer Center. And she has since then broadened her knowledge and skill in all aspects of clinical and translational cancer research through innovative training that includes didactic coursework with parallel clinical rotations. Her research revolves around the identification of biomarkers of resistance to CDK4/6 inhibitors and assessment of novel therapeutic strategies in advanced HR positive and HER2 negative breast cancer patients using single cell and bulk RNA sequencing methods. She also has studied cyclin E mediated immune changes in the mammary gland microenvironment during developmental and tumorigenesis using relevant transgenic mouse models to aim to use cyclin E as a stratifying biomarker for triple negative breast cancer patients that are susceptible to benefit from immunotherapy. Okay, so her talk today is called Low Molecular Weight Cyclin E Induction and Crosstalk with Immune Cells Potentiates Local Immune Response Leading to an Immunosuppressive Microenvironment at the Early Ages of Breast Tumorigenesis and Mouse Models. So everybody please join me in welcoming Sofia. If you would like, yeah.
Thank you so much for the introduction, Sarah. You're very kind. And good morning, everyone. Today, I'm going to talk to you on about my project on low molecular weight cyclin E induction and crosstalk with immune cells that potentiate local immune responses, leading to tumor formation at early stages of breast cancer in mouse models. Let me now talk to you shortly about cyclin E, which is a very well-known cell cycle protein. In normal cells, CDK2 cyclin E complex is tightly regulated and triggers the onset of S phase. However, in tumors, cyclin E is undergoing proteolytic cleavage by neutrophil elastase, generating low molecular weight proteins that have been found to increase tumorigenicity in vitro and in vivo. Now, in breast cancer, high cyclin E has been correlated with worse outcomes independent of subtype. And this is something that we recently showed in one of our publications as well, where we collected 190 treatment biopsies of breast cancer patients of different subtypes and that were receiving neoadjuvant chemotherapy, and we evaluated cyclin E status in their tumors. So what we saw is that cyclin E positive tumors had worse RFS, DFS, and OS compared to cyclin E negative tumors. Using the same cohort of patients and biopsies, we further assessed the tumor infiltrating lymphocyte status, and surprisingly, we found that high TIL tumors are also characterized by high cyclin E positivity, especially in the triple negative breast cancer cohort. Now, we know that cyclin E alone is a bad prognostic indicator, and tumor infiltrating lymphocytes is a good prognostic indicator for breast cancer. So we next ask what is happening when we're having high TILs in a tumor in regards to the cyclin E status. And surprisingly, we found that among the TIL high group, patients with positive cyclin E tumors had worse probability of pathological complete response compared to cyclin E negative tumors. So the overarching question is why tumor infiltrating lymphocytes in tumors lose their protective effect when cyclin E is overexpressed. And since we are talking about triple negative breast cancer mostly, we all know that pembrolizumab plus chemotherapy recently received FDA approval last year for both metastatic and neoadjuvant setting. Now, PD-L1 was used as a biomarker to stratify patients to predict response in the metastatic setting. However, new data from ESMO this year showed that PD-L1 cannot predict recurrence and differentiate responders from non-responders in the early stage setting. Unfortunately, PD-L1 is the most widely used biomarker of response to immunotherapy. However, two different IHC assays are being clinically used that have different ways of quantification and lead to inconsistent results. So there are not really established biomarkers of response to immunotherapy for the early stage breast cancer cohort, and there is a clinical unmet need to select the optimal patient population who will benefit from immune checkpoint inhibitors. Based on the above, we hypothesized that epithelial cyclin E expression alters the immune microenvironment pre and post tumorigenesis and can be used as a screening test to identify patients who are likely to respond to immunotherapy. Under this hypothesis, we first aim to evaluate the role of cyclin E expression in regulating immune responses at the mRNA and protein level. Second, to assess the preclinical treatment responses of immune checkpoint inhibitors in low molecular cyclin E high tumors using our transgenic mouse model that I'm going to talk to you about shortly. And last, we are aiming to validate our results in the future using human breast cancer tissues that express cyclin E before and after treatment with neoadjuvant chemo and ICIs. So for this study, we generated a genetically engineered mouse model capable of expressing low molecular weight cyclin E under the control of an MMTV promoter. So this sequence is inserted downstream of a TET operator, followed by an iris luciferase reporter, and P53 heterozygous mice, male mice, were crossed with P53 wild-type mice to then get inducible MPT, we'll be calling them from now on, mice that are also P53 plus minus to recapitulate the MBC cohort of patients, where human low molecular cyclin E is expressed after doxycycline administration in the drinking water of the mice. We tried to we made sure that cyclin E is specifically expressed in the mammary gland of mice that were receiving dox and not of the mice that were not receiving dox at the mRNA level, IHC, using luciferase activity measurements and through Western blots. And for this study, we generated age-matched groups of mice that received doxycycline for 3, 6, 9 months, and age-matched controls that didn't receive doxycycline. Continuous cyclin E induction causes tumor formation after 9 months in about 60% of the mice, with the median tumor latency being 10.5 months. So we collected mice at each respective time point. We isolated mammary glands and tumors from the mice that had tumors, as well as other peripheral organs. And we subsequently performed multiplex immunofluorescence, single-cell RNA sequencing, and flow cytometry. We also used an independent cohort of FEB normal mice, non-transgenic, that are not expressing cyclin E as additional negative controls. So we first designed a panel, a multi-marker flow cytometry panel that consists of different basic, I would say, populations of innate and adaptive immunity. And based on some changes that we observed in the T-cell populations, we further designed a more specialized T-cell panel that addresses more, let's say, T-cell differentiation populations, as well as Tregs, PD-1, and PD-L1. After integrating the flow cytometry data of 27 transgenic and 23 non-transgenic mice for both panels, and Sarah helped for that a lot. We revealed distinct cell subsets of innate and adaptive immunity, and interestingly, in the T cell, CD8 populations clustered in a different space compared to the CD4 populations. So we first asked, are there any age-related changes that are normal and belong to the normal developmental procedure of the mammary gland between normal and transgenic mice? And you can see here visually that there are some changes that we further quantified later on. The second question we asked is cyclin E driving any specific changes, or the changes that are that we observe are not cyclin E specific? So again, by comparing different mouse groups, we observed visually some differences that we further quantified. In general, we followed the following flowchart to come up with the cyclin E specific temporal changes. We first rule out age-related changes, then doxycycline-related changes. Then we came up with the cyclin E-driven changes, and we compared them over time to see which one of them are statistically significant over time. So what we found, by taking only mammary glands of 3, 6, and 9-month mice that received and didn't receive doxycycline, is that over time, monocytic populations are significantly dropped, whereas CD8 and CD40 cells are increasing, with the most significance we found in the 9-month time point. We were also observed some alterations in the T cell differentiation populations, with CD8 naive cells increasing. So that tells me that there is a naive T cell expansion that leads towards a more immunosuppressive phenotype, as Tregs are significantly increased over time in the mammary gland. So this is all before tumorigenesis. So we next addressed our questions by designing 2 additional panels of multiplex immunofluorescence containing these markers on the left. And we noticed that over time, there is a significant increase in vimentin, epithelial content, and TMT, that it's the double pancytokeratin vimentin positive cells. By designing, again, a more specialized T cell panel, we noticed that there is an induction of Tregs in blue and proliferating CD40 cells, showing again that there is an increased T cell activation over time. Next, we asked what are the changes that lead to tumor formation. So we compared the mammary glands of the mice. I don't know what happened here, sorry. There's something missing. We compared the changes in the mammary glands of mice that received doxycycline to those of tumors that were formed after 9 months. Again, we found that the decrease in monocytes was one of the reasons that led to tumor formation, with monocytes remaining stable after 9 months, so right after tumorigenesis. These cells were increased in tumors compared to the 9-month mammary glands. CD8 cytotoxic T cells were almost depleted in tumors, but CD4 levels, while increasing in the mammary glands, they retained significantly high levels in the tumors of our mice. We also noticed some changes in the T cell differentiation populations, with an interesting increase in Tregs in the tumors that is much higher compared to the mammary glands. To make sure that what we are seeing is cyclin E specific and not tumor specific, we compared our tumors with another breast cancer model, which is called PYMT, that is not expressing cyclin E. And we found that T cells, CD40 cells and CD8 cells were much higher expressed in the cyclin E positive compared to cyclin E negative tumors. The most important observation that we had is that Tregs were very significantly increased in cyclin E positive tumors. And the same happened with PD-L1, addressed in the CD45 negative cell populations, as well as the PD-1 PD1 positive CD4 positive and PD1 positive CD8 positive cells. So to sum up, we have a model, and we believe that epithelial low molecular cyclin E expression in the hyperplastic mammary glands before tumorigenesis causes some microenvironment reprogramming, which is mostly affecting the CD4, CD8 Tregs and monocyte populations, maybe through some DNA damage repair mechanisms that are yet to be identified. And this causes forms cyclin E hot tumors that are characterized by high CD4, Tregs, PD-L1 expression, suggestive of good responses to immunotherapy. So what we believe is that cyclin E can be used as a stratifying biomarker to identify triple-negative breast cancer patients that are susceptible to respond to immunotherapy. And with that, I would like to thank my lab, the QMRC Hunt lab in MD Anderson Cancer Center, and my Triumph CPRIT advisory committee, as well as our collaborators from ACSF core, especially Sarah and Karen, and the CPRIT single core directed by Nick Navin that helps me with the single cell analysis. And thank you all for your attention.
In the tissue, you could see if the Tregs are in close relation with, I don't know, some proliferative cells, like B cells, for example.
You mean using the multiplex immunofluorescence data? This is a very interesting question, and I need to look at that because I didn't look at the localization of the immune cell subsets, only the quantification.
I think that is important.
Correct. Mm-hmm. Yes, so it would be very interesting to look at the Treg localization regards, to other immune cell populations, definitely.
And the Tregs also express PD-1, like in high levels, or you could?
So I didn't I haven't added the PD-1 marker in the multiplex immunofluorescence data, but I definitely can quantify the results here since I have both PD-1 and PD-L1, right? So I can look at the PD-1 positive, Treg positive levels. However, if you see my PD-1 expression in general, it's pretty low, I would say. The total PD-1 levels, for example, in the CD45 positive fraction were a little lower. So since, I'm not sure if this would give high percentages when I localize the PD-1 plus Treg markers. But yes, I can look at that. Definitely. Thank you for the suggestion.
All right. Thank you so much.
Thank you.
All right. We're gonna move on as there's no online questions that have popped up yet. Would you care to introduce the next speaker?
So it's my pleasure to introduce you Anne-Laure. Anne-Laure, we were classmates together in undergrad, and we lost path, and flow cytometry bring us back together. So Anne-Laure is right now a senior research engineer at Toulouse Institute for Infectiology and Immunology. She did a master's degree in biotechnology and bioengineering in the University of Toulouse as well. She has more than 10 years experience in flow cytometry, and she's working in one of the big flow core in France, which is the name is the Influx core, and she's part of the French Association of Flow Cytometry as well. Welcome, Anne-Laure. And she's going to talk about mapping the human and murine immune system on Symphony 30 parameter cytometer.
Hello, everybody. Thanks, Nicola, to introduce me. So I hope you see my presentation. Yeah. So I would like to thank the organizing committee to have invited me. And I'm really glad today to present you the implementation of optimized and shared panels and the impact that we could get thanks to the common work that we have achieved. And I really would like to highlight the importance of putting our means in common. And I am illustrating this with the mapping of the human and murine immune system on the Symphony 30 parameter cytometer. So in the first part, I will briefly introduce you the context of this project. And in the second part, you will indeed see our collaborative approach. So I am briefly introduce you our institute. So Toulouse Institute of Infectious and Inflammatory Diseases is an immunological research center with 14 research teams and 4 technical facilities that represent 285 people. And on the flow course cytometry, we are 4 people, and our activity is mainly dedicated to our research center. Infinity, Restore,
Excuse me, Anne-Laure, if
Yeah.
I can ask you to put you in the slide view, the presenter mode in the PowerPoint.
Okay, sorry, excuse me. It works yesterday, so I restart the spot. Okay, I don't have I have entire screen or presentation PowerPoint, but I don't have entire. Is it okay?
We are seeing your small slides on the left-hand side. So
Okay.
if you can get the presenter view.
Okay, sorry.
Slideshow.
And like this? No?
No. It's good. It's okay. Just continue. Thank you.
No, I don't have in the, so I try again, but when I share my screen, I don't have diaporama. So I don't know why, because yesterday I have it. I have only a presentation FlowTex PowerPoint. I'm really sorry.
No problem. Just share your screen again and we are good to go. Thank you.
Okay, I'm really sorry for that. So Infinity, Restore, CRCT, I2MC, and IPBS, we are all working in the same network with our expertise, our means that we share throughout this network, and TRI, Toulouse Réseau Imagerie. This is what we are in this network. So now let's jump to our context of our project named Octopus. So Octopus is a major regional project. This is an integrated project that brings together academic and private structures to develop new multimodal characterization tools in the context of aging. For the academic part, 2 technological platforms or life sciences are involved, TRI and MetaTool. And for the private part, there are 3 partners, Pierre Fabre, BD Biosciences, and Rigor. Thanks to this private partnership, we have acquired a Symphony cytometer, and BD Biosciences help us to design panel and to fund a temporary contract. So what is our aim? We want to map the human and murine immune system, and to do so, the cytometry appears to be a reference technique to determine the phenotype of diverse populations, to collect detailed data on the immune response, to study rare cell populations, and to isolate populations of interest by cell sorting. This technology enables to finally characterize complex populations, to work on a large number of cells, and thus to obtain robust statistical results. The scientific objective of this project of this project is to phenotypically characterize the networks formed by the cells of the immune cells in different cases at the extreme stages of life, in pathological context before and after treatment, and in response to vaccine and infectious. Yes.
Sorry,
there is a problem.
Yeah, it's not advancing your slides. Maybe you can try.
Maybe I can try to share my screen. It's easier.
Just share the screen. Maybe.
Yeah, I think I do that. I'm really sorry because yesterday it works.
I know. Yeah. Let's see.
So, okay, I entire screen. I hope it works. Okay, do you show my slide?
Yeah, that's better.
Yeah.
Okay, thank you.
So
Sorry.
I'm really sorry. I don't know. I don't have the PowerPoint diaporama in the selection. I don't know why. So I continue. Sorry. Okay, I finished this map. So what is our goal for this project? We want to identify predictive biomarkers and therapeutic targets to prevent or correct age-related abnormalities. And to do so, we will perform multidimensional protein expression analysis by flow cytometry of different subpopulations of the immune system, like innate cells, T lymphocytes, B lymphocytes, to compare frail to elderly people. So now you have understood the whole collaborative frame of our network. I describe it further, the work of our facility with our approach, our steps, and achievements for this project. So we decided to set up a structuring and mutualized approach for the creation of common core panels. And to take up this challenge, we have interacted with our private partner, BD Biosciences, and we have created within our center a symphony working group with a referent power team and the 4 people of the facility. And these exchanges have enabled the development of 4 common panels that represented 150 antibodies. And the objective was a real challenge, but because we want to have a real collective approach for the benefit of our users. So initially, there was a strong collaboration between the core facility and the Symphony Working Group. Each one brought its expertise, scientific, technical, and technological. And this approach of combining and sharing all skills is crucial to develop a structured and standardized approach for the different steps from the panel construction to the panel validation. So we have set up a workflow, which you can see here, the different steps from the cytometer validation to the cytometry store. And you can see the involvement of the different partners. And you can see that we have a collaborative approach with shared expertise throughout the project. So the first step was to validate the sensitivity of the cytometer. And for this step, the cytometry core facility worked with BD, and we want first to establish the spillover spreading matrix of our symphony. And for that, BD Biosciences provide us anti-human CD4 antibodies coupled to 28 different fluorochromes, and the core facility did the titration, filtration, and analysis. And you can see here our spillover spreading reference matrix that we use to design all panels. So the second step was to design panels, and this step is essential. There was a real collaboration between the cytometry core facility, BD Biosciences, and the Symphony Working Group. Everyone was able to bring their own expertise. And the step was a real exchange because we are complementary, and together we are stronger to meet the needs of the researcher while taking into account the specificities and constraints related to the panel design. So here you are an overview of the immune cell study by Infinity teams. The team works on lymphoid and myeloid cells, and the majority of the teams work on T lymphocytes. And I think this overview is important to understand why we have chosen to set these panels and also for the selection of the markers for these panels. So to design panels, the first step was to define the nature of the panels. We first had to choose which species to work on, which cell types, and especially which subpopulation and which markers to choose. And and after many exchanges, it was decided to design panel both in murine and human, to work on PBMC, spleen, and lymph nodes, and finally to design 3 panels, the lymphoid panels, the myeloid panels, and the functional panels. The functional panels was quickly put aside because with the choice of the selected marker, there were strong constraints related to the intracellular staining protocols which were not compatible, and we decided to focus only on the lymphoid and the myeloid panels. So the second step was to build the panels, choose the marker and how to build it. And after, again, a lot exchanges, the panels were all built in the same way. For the choice of marker, we select lineage to identify the major population, secondary markers to characterize the subpopulations, and finally, activation, exhaustion markers to compare treated and untreated samples. For the construction of the panel, they were also constructed in the same way. We choose to add a variability die, use barcode to analyze several samples at the same time, make a core panel, have options to characterize more thinly subpopulation, and finally to keep a free PMT. So in this way, researcher can add its favorite antibodies. So now I will present you the next step with the murine lymphoid discovery panel. And after choose the nature and how to build our panel, we have to select markers. And this step took time because it was necessary to get everyone agree, and it's not easy to make everyone agree when where you have 18 people in the same room. But at the end of the process, we obtain a consensus on 33 antibody plus the live dead, and you tell me you have a 30 parameter and you have 34 parameters, so we have to deal with that, and I show you the solution. So finally, for the mouse lymphoid panel, we have a core panel, 2 options, and 1 free PMT. So for the core panel, we have the live dead die, we have 2 antibodies for barcode, and we have lineage markers, B-cell differentiation markers, T-cell differentiation markers, and activation and exhaustion markers. For the core panel, you have 22 antibodies plus the live dead. We have 2 options, 5 antibodies for the Treg option and 6 antibodies for the B cells options. And we also made the choice to keep the GFP detector totally free from core and options to enable researchers who have a GFP reporter gene in their mouse model to use these panels. So at the end, you can use only the core, so 23 parameters, or the core plus 1 option, and you have 28 or 29 parameters in your panel. So the next step was to develop the fluorescent cell barcoding and titrate antibodies. So as a reminder, fluorescent cell barcoding is a high throughput multiplexing flow cytometry technique. It uses fluorescent probe less or more concentrated to stain different samples. And this is what you can see here. Each sample has its own specific signature or fluorescent barcode. Once watched, the samples are all put in the same tube to make a single antibody staining. And you have only 1 tube instead of 4 to acquire to the cytometer. And the interest of this technique is multiple because it enables to have at the same time an homogeneity of the staining you can reduce the antibodies consumption, you can reduce your acquisition time, and you decrease the inter-sample variability to get robust results. So in our panel, fluorescent cell barcoding was developed with antibodies and not with fluorescent probes. We use anti-CD45 antibodies coupled to 2 fluorescent fluorochromes. We choose to do 4 conditions in order to analyze 4 samples in 1 tube. And for this, we tested serial dilutions of 2 antibodies, and we choose dilution, allowing us to separate correctly the samples, but also to be saturating. And you can see here the results. We can analyze the 4 samples in the same tube without any problem. So we also titrated all the antibodies, and for the murine lymphoid panel, it is represented 33 titrated antibodies plus the live dead. And to choose the optimal dilution, we calculated the stain index of each dilution point. And as you know, the higher the stain index you have, the better separation between the positive and the negative population you have. So the next step was to test the panel. And for this step, we first tested the core only with immunized mice. And the objective was to optimize voltages, the composition matrix, and validate staining and validate the immune populations. So first, we want to validate if the dilution we had chosen with the titrations were optimal when testing all the antibodies together. And unfortunately, it was not the case. It doesn't work at the first time. We had problem with 3 antibodies, the NKp46, CD127, and CXCR3. And as you can see here, for the NKp46, we increased the antibody concentration from 100 to 50, and this solved this problem. And we apply the same approach with the 2 other antibodies, and that solves the problem too. So when the core panel staining were validated, the next step was to validate the lineage population as well as the different lymphoid subpopulation. So you can see in this UMAP representation that we succeed in differentiating the major populations such as TCD4, TCD8, TCR gamma delta cells, NK cells, and B lymphocytes. So when then we check that we have also found the different subpopulation of B lymphocytes and CD4 and CD8 T lymphocytes. And you can see on the left UMAP that we can distinguish the different B subpopulations, B subpopulation such as marginal zone B cells, plasma cells, and follicular B cells. And on the right UMAP, you can see that we succeed to well define the different T subpopulation, T helper cells, and T cytotoxic subpopulation cells. So next, after validating the core panel, we have to test the core with the 2 options, with Treg and B cells option. And all the optional antibodies works well. And you can see here the results obtained for the 5 antibody in the Treg option. And there is no problem to analyze the population of the Treg population. So the goal of this project is not only to mutualize the panels developed, but also to make them easily accessible. So to do this, in parallel with the panels, we are setting up a flow cytometry panel stores. This is a tool that allow us to manage antibody stocks, orders, and invoicing the quantities of antibodies delivered. And we have decided in a collegial way that the cytometry core facility would manage a stock of antibodies in order to make the 30-color panel accessible to the teams who use them in routine as well as punctually. And from a financial point of view, this procedure is really advantageous for the teams. So this is the end of my talks. So we have two kinds of prospects, short-term ones and long-term ones. And the third thing we have to do is to finalize the lymphoid panels. We have to make available the murine lymphoid discovery panel to antibodies panel store. And we have to finalize the human lymphoid discovery panel. The second one is to apply the same structuring and mutualized approach for the development of human and murine myeloid panels. So the final goal is to provide with 4 standardized, ready-to-use panels from the staining step to the final analysis. And at the end, this collaborative approach made available a 30-color panel, not only for our institute, but also for all the Biosanté community in Occitanie. So I really thank again the organizing committee to inviting me. I would like to thank my colleague who works with me on this project, Hugo, Valérie, and Fatima. I would like to thank also the BD application scientist Thierry Langlois, who helped us to develop these panels. Thank you for your attention. I am really sorry for the virtual with the problem with my PowerPoint. And I'm ready to answer if you have any questions. And sorry again for the problem with the presentation.
I think we're going to address the questions in the roundtable, and we're going to get the next speaker lined up.
Okay, so our next speaker this morning is Stephanie Widman. Stephanie has more than 20 years of experience in flow cytometry reagent development, assay development, and single-cell multi-omics site-seq applications. Her early work in the field includes polymer development for use in flow cytometry, which helped drive the expansion of high-parameter flow. Currently, she's the Associate Director of Scientific Affairs at Becton Dickinson and is leading a team of scientists with diverse expertise, generating materials that help serve as important educational resources for researchers to improve their understanding of flow cytometry. The title of Stephanie's talk is A Strategic Design Approach for the Development of an Optimal Backbone Panel. Welcome, Stephanie.
Thank you very much. Make sure that you can hear me, and I am presenting. Okay. Good on sound, and you can see it, right? Okay.
Yes.
Thank you very much for that introduction. Today I'm going to share with you a strategic design approach for the development of an optimal backbone panel. And there we go. And we all know that flow cytometry has evolved immensely over the last 20 years. Over 20 years ago, it was common for publications to have panels with 3, 4, 5 color panels. And now we're looking at publications with 28 markers or more to interrogate their cells of interest. But how do you increase the size of your flow cytometry panel? Today, we're going to review 2 different approaches that are used to design either a standalone panel or a backbone panel. Both of these approaches are going to rely on the fundamental rules of panel design, so fluorochrome brightness and spillover properties, and antigen density and co-expression of your markers of interest. So your standalone panel it's your project-specific panel. You have a particular need, you're going to design a panel for it, and it can be relatively easy to design if it's in the low order area, say 5 colors, because you can utilize the spectrally separated fluorochromes. But it can be very difficult to expand. You think, oh, I have this panel I made. Let me just add in 3 more markers. It shouldn't be that hard, right? Well, you're bringing on some challenges that we're going to review in a little bit. And then those challenges sometimes can be so complex that you may actually have to redesign the whole panel. On the right, we're showing a backbone panel. The concept is flexibility in the sense that it's designed with the idea to be expanded later on. What's more difficult about designing a backbone panel is that the challenge to the panel you face up front. So you are utilizing fluorochromes with a higher spillover or more spread into your backbone to potentially leave open the open channels so that you have the flexibility of drop-ins and minimal panel design for those drop-ins. So one of the first steps to designing any panel is to look at a TSM or total spread matrix. And if you're not familiar with a total spread matrix, it is very similar to an SSM, spillover spread matrix. This can easily be generated in FlowJo as well. You have the option for SSM or TSM. And what TSM does is it removes the brightness out of the equation, which allows you to compare the red boxes, I say, from what you see here. So if I want to look at a red box spread in this UV area of 2 fluorochromes, the red spread is comparable to the red spread that you might be seeing over here in APCH7. So I'll walk you through a little bit. We've got BV711 here, and when it's used with BV480, you can see it's a green box, and green boxes are great. It tells you that there is minimal spread. You can see that the spread of the positive is about the same width as the spread of the negative. As we move along, we get over to BV650, and we can see that we're starting to get into the territory of spread, and which means that you are going to have to design your panel potentially around this spread. But the red boxes are just significantly worse, right? You can see here that there's dramatic spread. When there's a red, this type of spread, often it means that you're going to try to put the fluorochromes on non-co-expressed markers, and just more advanced panel design, more time taken for optimization. So if you start out making a standalone panel and you look at the TSM or SSM, you pretty much are thinking, I'm just going to go right to those green boxes because I'm not going to have to worry much. And it's actually a great concept because you have some very well-performing fluorochromes here. There's a lot of availability for these, for your markers. And so that's why, in a way, this becomes rather easy to design a low-order panel. But that concept of then adding on, taking it from 5 to 8 to 10, now you are fixed with your initial 5 that you selected. That's what I'm showing over here on the right, is that if you select these 5 to start, you now have the remaining ones to choose from, which means as you look at these fluorochromes that are available, you have to think about how they're going to impact the ones you've already selected. And you can see that you're starting to get into some red territory here. Not only do you have to evaluate how these fluorochromes are going to impact your standalone panel, you now have to think about how they're going to affect each other and the spreading that occurs between those fluorochromes. So while it was easy to start off, you have that kind of exponential difficulty in adding on to this panel. And as I mentioned a little bit, that you're kind of using a lot of the more higher percentage of in catalogs or available fluorochromes. So for example, PE, if you're adding on a marker and it has limited fluorochrome offerings, chances are some of those offerings are going to be FITSI, PE, maybe BV421. And so you're going to be forced to go into your additional potentially red spreading fluorochromes. Also, this panel is not conducive to adding in a GFP, fluorescent protein, or using something like a streptavidin PE for the detection of antigen-specific cells. So now we'll review the requirements for a truly flexible backbone panel. Number one is that the backbone panel, it has to be of good resolution, good quality. You have to be able to identify your populations of interest. That is key. Then once you identify that, panel, that backbone panel, you have to make sure that the panel minimally impacts those channels that have been identified for the drop-ins. And then you have to check that those drop-ins don't impact your backbone. So 2 and 3 are pretty much the vice versa, that backbone panel doesn't impact the drop-ins don't impact the backbone. And lastly, how we mentioned that extra critical part is that the fluorochromes that you picked for the drop-ins don't impact each other. So we decided to test out this strategy with a human T-cell backbone panel for a 3-laser instrument. And you'll see here the markers that we selected through CD3, CD4, CD8, CD45RA, and CCR7. And then over here on the right, you'll see that we've selected the spectrally separated fluorochromes to be the drop-ins. So that is your like BB421, your FITCs, type blue excited fluorochromes, your PE, and your APC. So going back to the backbone over on this side, I'm showing you the TSM for these fluorochromes. Because you are designing the backbone, that's where you get the option to work around some of that spread that you see from the matrix, the spread matrix. And because we have CD4 and CD8, you're able to manage some of that spread, as well as having a lineage marker where their antigen density is to the level that the negatives could probably take some spread, and you will be able to resolve your population. So this is where it's time consuming, slightly challenging, but depending on what your biology is, this is where you are able to use those fluorochromes that may not be ideally spectrally separated. Okay, so we designed our panel. Let's see what the panel looks like. Here, the panel is on 2 different configuration of flow cytometers. We have a 3-laser 12-color and a 4-laser 16-color. And you can see our gating strategy of CD3, CD4 versus CD8, and then the CD4 and CD8 gates looking at CD45RA versus CCR7. You can probably tell in the middle plot that there is some heavy bi-exponential scaling here, but these are not co-expressed. So as long as we're able to gate these populations, they can take some of the spread that could be from the other reagents in either the backbone or potentially from the drop-ins. And then you can see here on the right, we have our more critical populations gated. These are gated by FMO. So, and as you can see, this is a very good level of resolution for these cells of interest, and meets that first need of a good resolution for your backbone panel. So now that we've designed a good backbone panel, we want to see if the backbone panel impacts those channels that we've identified as available for drop-ins. So to do this, you can see we have these histograms with unstained cells in each of the channels. So here are our open channels that we identified was BV421, FITC, PE, and APC. And then, so the red population are unstained, so no fluorochromes, where the blue cells have the backbone staining on them. And you can see that there is a minimal spread into these open channels. And as an example of what's not necessarily recommended is just to see what spread really does look like when you look at them this way, is that you can see that when the backbone panel is stained on these cells, you are getting a spread effect into this channel, which could impact the resolution of whatever marker you choose to put in 605. Down at the bottom here, we have the TSM, so you can see clearly that it's the BV510 that's causing this spread. And another way to visualize that we have here on the right, which is CD3 BV510 into 421, one of the recommended drop-in channels. And then we're showing BV510 into 605. So here you can visualize the spread specifically from the 510, where here you just know that it's coming from the backbone panel. So these are the recommended drop-in channels, which then meets that criteria of the backbone minimally impacting those open channels. So now we're going to review how the drop-in fluorochromes can affect the backbone. So to do this, we decided to add the fluorochromes that are in the backbone, or in the open channel, excuse me, in CD45. So we figured, what's a high antigen density marker that we could use to demonstrate that what an effect could be if you used one of the highest antigen density markers that we have. So we added CD45 here. Sorry, let me explain the backbone panel alone, just the CD4 positive or CD8 positive cells. And then this bottom row, we have the backbone where we've added in CD45 BV421. And you can see that the resolution is held up. It's minimally impacted by BV421 on 45. You might say you wouldn't ever do this. You'd never really put 45 BV421 on CD45, but this is just a case to show what if someone did use it on a high antigen density, and it ensures that no matter what you pretty much assign under that level of antigen density, there will be no impact to the backbone. On the right is just the addition of the other three fluorochromes, the FITC, PE, and APC. And we're able to show all of the different populations that are gated in this backbone panel and their change of percent positive when those are added. So here we're showing no impact from the drop-in fluorochromes onto the backbone panel. Okay, and lastly, we talked about that step where you have to make sure that your add-on fluorochromes don't interact with don't impact each other. And because we pretty much flipped this, right? We took that standalone panel concept of starting with the best ones, we're kind of ending with the best ones. So by using those fluorochromes as the drop-ins, we already know that they have minimal impact or spread between them. So to demonstrate that, you have again the spread matrix, but then also down here, just for visualization, we have CD45 PE into 421, FITC, or APC. So these represent the 4 open channels. And then again, as an example to show what's not a good or recommended open drop-in channel, we have CD45 PE into BB605. And you can see if you had this, now you're bringing on the fact that you're going to have to redesign or work around this spread. So I was able to share with you the data to show that this backbone panel really is flexible in the sense that it can have any 4 markers dropped into it as long as they are in those fluorochromes that are in those open channels. So now it's kind of more the fun part where you get to see the applications of this panel, where we played around with different different protocols, different panels to see how the panel performed. Firstly, we thought, hey, let's just run this on a 5-laser instrument. And then if we have a 5-laser instrument, that means that we kind of get an extra drop in, because we know that BUV 395 is not going to be impacted or impact the backbone panel. So on a 5-laser instrument, there is an extra drop in that's available. And then I didn't talk about this, but viability dye is kind of also a wild card because it's excluded from the panel. So here we are able to have our 5-color backbone. We dropped in the 4 plus the 395 and then the viability marker. So this is a 11 marker panel. And let me show you then actually the biologies on the next page. So here, this gating strategy I shared before, followed with being able to identify TSCM and naive with CD95 versus CCR7, and then those cell populations interrogated for KLRG1 and CD127 on this row, and then CD27 versus CD28 on the bottom row. Another experiment that we wanted to do to test this out was a standardization experiment. We set 3 instruments up with the same application settings and then ran the panel. In this case, it was a CD4 TH subsetting panel. Same sample was run on the 3 instruments with their standardized settings, and we were demonstrated that that between the 3 instruments that was a good performance for a standardized approach to this panel. We also wanted to evaluate different buffer systems. So the FOXP3 or T regulatory cell panel we thought would be a great panel to demonstrate this. This panel uses the transcription factor buffer. In this case, we stained all the surface markers first, then fixed and permed, and then applied the FOXP3 staining. And you can see here our CD127 versus CD25 to then fully pull out the Treg cells with that are FOXP3 positive, then further analysis on CD45 versus HLA-DR for naive and effector, and then looked at the CCR7 levels on those. Another protocol that's pretty common is activating cells. So we chose an inhibitory panel for this, activated healthy PBMCs for 3 days with anti-CD3, CD28. Because these cells are cultured for 3 days and these cells are activated, it's not surprising that we see a little bit of change to the backbone panel. This is because the cells are now, being cultured and activated. You wouldn't be surprising to see up or down regulation in some of the markers of the backbone panel. And then here we're able to pull out PD1, LAG3, and TIM3 in our some in our resting, but then also for these other ones in our activated. Then just on the right here, we have some percent positive for CD8 or CD4 cells. So in conclusion, I was able to share with you today a strategic design for a backbone panel that is truly flexible and allows the expansion of recommended fluorochrome drop-ins without impacting the resolution. The panel design process for adding new markers with if they're in those recommended channels is simplified because you don't need to worry about spillover or spread or even antigen co-expression. They're not a concern. We still recommend matching fluorochrome brightness to your antigen density for optimal resolution of your cell population. And the number of drop-ins, it can change depending on your cytometer's configuration. And that I was able to share with you some fun data showing different applications of the backbone panel in different protocols. And I probably talked a lot faster this time than practicing, but that is the end. So thank you very much. And if there's time, we could do questions, or we can go into you can make up some time and go into the roundtable.
Thank you so much. We are going to go to the round table, I believe, and answer some questions unless, hold on, we lost our coordinator here. All right, so did Mark have a prepared talk, or is he just partaking in the round table? Okay. So, Celine, are you?
Hi, hello.
There we are. Thank you so much. And so Celine will be our moderator for this session, taking questions. We'll field some from the audience as well as some from the Zoom, from the YouTube stream. Thank you.
Yes, thank you for the organizers for providing this opportunity to lead this this roundtable, this discussion, and I would like to thank the speakers from the session for their presentation. We have heard absolutely brilliant talks, and it is incredibly inspiring to see how the community altogether is harnessing and transforming flow cytometry technologies into decision-making tools. Yes, so please, if the crowd or if the virtual attendants have questions, please post them on YouTube or ask them. Maybe I can start with one question of my own. So I've personally worked in very close collaboration with Cytec to do some extension of backbone panels. And so I would like to direct my first question to Mark, because we've heard very good talk about how to build a backbone panel. But now, Mark, could you maybe summarize for everyone, when you have a backbone and you want to extend it, what are the key considerations and the key steps that you ought to take? And is there a limit up to which at some point you say, okay, I better start from scratch?
Well, more often than not, you don't know what the backbone panel is going to, what the drop-ins will be into the backbone panel. And so one of the things you can do, and you saw this in the previous presentation, is to put things at the low end of the emission spectrum off a given laser, because spread is directional, right? And so, using BB515, PE, APC, BV421, even BUB395, though it's not as bright on some instruments, is safe. On a full spectrum machine, you also leave the adjacent detector empty. So you want to make sure that you're not dealing with co-expression at some point from that adjacent detector, the one up from wherever you're putting in the drop-in. If you design the panel when you're, as part of the backbone, and after a while of having designed, 3,000 some panels over the last eight or 9 years, you learn what the directionality of spread is for all the different fluorochromes. For instance, you saw in the last example, BV605 and PE. If you put a lineage marker in 605, then you can measure something safely in PE. So you learn those relationships over time. And when you're building your backbone, you just automatically know where to put things, whether it's a lineage or you're measuring something, right? And that's really the best way to move forward. For additional backbone markers, then that becomes a little more difficult. You can still, plumb the advantages of the directionality of spread. That's very helpful. But as you add more and more places where you're trying to drop things in, you have to be more and more careful. And it depends on, the number of lasers on your machine. Obviously, if you don't have a 561, you're a little more limited on your options than if you're running a lot more fluorochromes off of a 488. So those are the sort of things you just take in mind. And that after a while, it's also dependent to know, it's also good to know what lineages you're dealing with, how much co-expression you're dealing with, just in general, what the panel will be used for.
Okay, thank you, Mark. I can see that Sarah has dropped a question in the chat. Can you discuss designing protocols for including both fluorescent proteins in the backbone panel and intracellular staining without diminishing signal from either? Open question if you would like to tackle it first.
Okay, I guess I'll jump in. If nobody else wants to. So one of the things we did when I was at Q Squared is we created a database. And everybody should do this. There's a couple things you should do. You should build yourself a co-expression table that's exquisitely detailed, right? And it'll save you endless amounts of pain. and you build it over time. I think mine's up to like 1,000 different markers now, and it's granular down to level, maybe 20 different subsets. And, you know, somebody should actually, you know, publish this. It would be very useful. Could you repeat the question one more time to make sure I'm directing the answer correctly, so I don't get lost here?
So the question is about designing protocols that include both fluorescent proteins in the back, and staining.
Right. So first of all, start with a co-expression database, and then build a database of the optimal conjugates of every particular clone. So, for instance, you know, we learned this over time, and it's instrument specific to a certain extent, but generally it really is conjugate specific. For instance, there's a marker for CCR7, and it works well in PE, PECY7, BV421, and Alexa 647. And you would think, just out of the fact that APC is also so bright, that would work well, and you would see that it resolves with that conjugate as well. It turns out it doesn't, and it's not predictable always what will work and what won't. So you have to learn what conjugates will work. Then you then take those conjugates that are optimal for surface staining and see how they stand up to various perm conditions, right? So I was one of the back in the day one of the people who helped develop the Lysfix buffer that's used for whole blood. I actually did that with working with Todd Covey at Amgen and driving back and forth to Pharmagen and trying to find a formulation that worked. And what you have to do is, and there was a Martini project we worked on at the same time when I was at BD. And so we learned what markers were maintained on the surface. We learned what markers were impacted by the amount of methanol in the perm buffers. We learned a lot about, the conditions under which certain conjugates would work, such certain fluorochromes would be maintained. And a lot of this information, unfortunately, you only learn by experiment. It's not really memorialized very well anywhere. So you have to build your own database, and when you're working with the particular markers of interest for you or the people who use your core laboratory, you build this database, and over time, and you can say, okay, so if you want to look at CCR7 and you're running FOXP3, you can get away with PECY7 and PE, but Alexa 647 and BV421 won't work unless you can extract autofluorescence, and then you can also use BV421, but then that's machine specific, right? So you learn that kind of detail, and over time, everyone who works with you learns that sort of detail, especially if you share it in a database. So I would recommend everyone don't make everything a one-off. Make sure whatever you discover is memorialized somewhere and shared with as many people as you can. That's why I like these meetings, because everybody's getting together, and regardless of where we're coming from or who we're working for, we all have the same goal, and that is to make flow cytometry a useful tool. And the more we share information, reliable information, and not apocryphal information, the better off we all are.
And Celine, if I could add a little bit to that. Mark made some great comments there and points, and I think a lot of this of what we're coming into is a time where we really need to understand our fluorochromes and understanding their expressions or impacts into the different treatments that you do, and also how they all play together. For example, Cytec has some wonderful tools to look at things in terms of similarity. The Cytobank used to have a buffer impact chart, and again, building those up internally within your facility is great, but also I think it also creates a great opportunity where there are now probably over 140 different commercially available fluorochromes, and they get impacted by different types of treatments that people have, and thinking about building those types of databases are incredibly valuable. And then also, when people are working even with transfected cells or reporter-mized fluorescent proteins, again, tricks to kind of deal with some things where a buffer may impact GFP or other fluorescent proteins. A lot of times, sometimes people think, oh, you permit and things leak out of the cells, which may be true to some level, but also some of these buffers impact proteins, and so you just lose the signal itself, of which then some of these anti-GFP antibodies and similar types of antibodies are available to kind of enhance those signals to make sure you maintain that brightness. So again, I think it just is with the new capabilities of the instrumentation, knowing these fluorochromes as more and more come out, and then a community creating the tools to help understand how they can be used together, I think is going to be key for making cytometry easier going forward.
Yeah. So I'm glad you brought up the similarity and complexity index. And one of the reasons that I'm mentioning this right now is what I noticed was when I work with people, they design their panel, and at the end, they go and they look and see what the similarity and the complexity of the panel is that they've designed. And that's not the way to work. What you want to do is start with the number of markers you have and say, what are the optimal fluorochromes that I could use, if I can use them, if I can find the conjugates. So you build a fluorochrome set with the least similarity and complexity, and then you build your panel and see if you can design to that fluorochrome set. I think if people would work in that way, and it's keeping in mind that every panel is instrument specific, absolutely instrument specific, and that you're designing for a particular instrument. So choose your fluorochrome set well, and you'll benefit from that, and just avoid, however you attain that information, build it to have the minimum amount of spread and similarity between fluorochromes, and you'll have a good starting point.
Right. So that question, which was about intracellular staining, I'd like to actually come back to Anne-Laure, because if I'm not mistaken, you mentioned in your presentation that you had initially planned on having an ICS panel that in the end you choose to drop out. Could you explain a bit more why it was dropped out? I think it might be interesting for everyone to understand what were the specific challenges that you faced with this panel that you decided to drop it out in the end.
Are you sure to understand why I put out the GFP panel? The GFP detector?
Yeah, I think you mentioned you wanted to have an intracellular panel, or like one of your backbone should have
the functional, why we put out the functional panel. Because we have a lot, because we want to use the complete cytometer detectors, and we have a lot of intracellular staining. And when we look at the protocol to analyze this protein, they are very different, and there is a big challenge to design this type of panels. And the priority for us was to focus on surface markers, because for us is a new technology to go to 30 parameters, and to start for us it was easy to start with surface markers. That's why we put aside the functional. But I think when we have finished with the two surface markers, myeloid and lymphoid, we think about the functional.
All right, thank you. Now a question from Kathy. What are your opinions for adding in antibodies that do not come pre-conjugated? For example, if you're using novel dyes, such as the Nova Fluors or something, and you have to do the conjugation yourself in-house, how do you add these to your backbone panels? What is the best way to proceed? Maybe Stephanie, would you like to take the question?
Sure. Well, first off is just testing the performance of those custom conjugations, right? Doing your a titration for optimal staining concentration. If you're in an instrument that has additional parameters that are not affected by your backbone, then I could see that those potentially would open up additional drop-in channels. A lot of the Nova Fluors, for example, are in spaces that are tighter, or and may work well with a backbone panel. But ideally, it's making sure that you're getting the resolution that you need before you even look at it in the panel.
All right, thank you. Then we have a question from Nicola. Because the panel design is instrument specific, how to design a panel for multi-site study?
Well, you have to assume that your instruments are standardized, right? First of all, what you can do is, you could take a measurement of all your instruments and create a virtual predicate and say, you know, this is how the majority of my instruments will perform. You know, you can work backwards from that standpoint, and that's one of the ways you can standardize a large number of laboratories and a large number of machines. But if you start with standardized machines, then, you know, that's the best starting point. As long as they're all the same platform, they all have the same lasers, they all have the same filters, right? Then you can safely design for them. But if you're designing a panel for multiple instrument platforms, it becomes much more difficult, and you have many fewer options, and the complexity of the panel has to be reduced to accommodate that. And so the best answer is make sure you have the same instrument everywhere in a study. And if you don't, then you have to look at the individual performance of each, see what the limitations of each, and then impose those limitations on the panel design as a whole, which is a terrible thing to do. But, you know, people do that because they have no choice. Right?
Okay, Nicola has another very great question. In your opinion, how far is the field to have standardized backbone panel available? And I would go one step further. How far are we from having lyophilized standardized backbone panel available, that we just need to add ourselves to the tube and not having to prepare a backbone mix anymore?
Well, my thought on that is one of the hard parts of the backbone panel was agreeing on what markers should be in that backbone panel. So as you'll see, as you saw in ours, we were down to 5, where ideally people would think, oh, I might want CD25, or maybe I want to include, Tregs into my backbone as well. And we started to get all these options. We're like, no, we need to go back and give the flexibility to the researcher of how they want to customize the panel. So that kind of goes then with the dry down or a lyophilized solution, is that it's either going to be very specific for a target customer, or it's going to be limited so that it can be broader so that more people can use it. I personally see kind of a place for both. Maybe in CROs, they have the need for large studies, and so therefore they can really get a customized panel. But for a researcher that might want the panel one day to look at these 4 markers, and then next month it's for another 4, then that more simplified backbone is probably the solution.
Right, thank you. The next question is directed to Anne-Laure. How challenging was it to define the panels, considering you were targeting cells from different tissues?
It's an excellent question. So it's a real challenge to define the panel, considering staining cells from different tissues, and the role of the collaborative approach is very important for this step, because we can lean on the expertise of the researchers and on BD also experiences to define the panel and create a protocol for the different tissues.
Mm-hmm. Right, I think that's also a bit what Stephanie was saying, that if you are working with a very specific sample type, then the backbone might just not work as well as when it was created. Yeah.
Yeah, one of the biggest problems you run in when you're running different tissue types is the different autofluorescence signatures you're going to see for each of those tissues. And, taking that into accommodation really limits the panel design. One of the nice things about full spectrum flow is you can extract that autofluorescence. And I think Diana will have a talk later on, I believe, in the session where she addresses that. And I think that'd be very useful to see how, you can deal with autofluorescence in various tissues.
So following up on this remark, and disclaimer, not as a mean to oppose technologies at all, between spectral flow cytometry and conventional flow cytometry, is there any advantages, inconvenience that make backbone panel design or backbone extension easier, harder on one? Do they share the same limitations? What are generally your thoughts? Because, well, we have Mark working on spectral, and we have Aaron, Stephanie working, and Anne-Laure, I understand, working more on conventional. So
Well, I've been doing this since 1979, so over 40 years.
I think Mark has been working with both.
For a long time. And so what I'll say is, you just design to the instrument you have. Most often you don't have a choice, especially if you're using a core, right? And you say, okay, I know how this instrument performs, how the markers will perform on this instrument, and you design to that instrument. Some instruments have particular advantages. Every instrument's a little bit different, and you adapt to the instrument. I mean, having additional tools is nice, but that doesn't mean it limits your work as long as you know the parameters and the limitations within which you work. It's that simple. I started on a machine that had no log amps, 2 fluorochromes, and no computer. Had a pulse rate analyzer, and you had to take a picture of an oscilloscope to save your data. It was, the data was terrible, and the work was frustrating, and it took an hour and a half to set up the machine every morning because you had to align the lasers every day. So, what we all have now is just absolute luxury, right, compared to what people had years ago. So, just work with what you got.
Right. And going back to some of the, Mark made a comment about, the lower emissions off of different channels, off different laser lines. And, I generally kind of refer to it as working with the bookends. And even though the far red, you may run into a few more problems as spread goes up, but you can kind of stagger those a little bit. But again, as you have more lasers, for example, and things like that, like a lot of these rules are still the same. And whether it's one type of technology or another, and working within that technology. And, I think it was great that Anne-Laure talked about cell barcoding. Again, like using your instrument for the capabilities that it has to try to get more out of it, and being smart in ways that you're trying to use what it is that's available.
Yeah.
Yes. Okay, next question from Mikey. Can you talk about the clone selection process for the backbone panel?
Yes, I can. So in general, we went with more published clones. So honestly, for CD45RA, we had a couple choices. For CCR7, we had choices. So we had choices for everything. So there was quite a bit of thought that went into those. If at one point we were thinking about using a different CD45RA clone, and at the end we decided to stick with the HI100 because it has more history. So as you'll see in those particular ones, there are more historic, except for the CCR7, which is a new clone from BD that gives the high resolution. So that would be the only the difference there. There was a little bit of concern on CD8. SK1 is another clone that a lot of researchers use. But ideally, if you used different clones, you would just need to check that the performance of that panel, so that first checkbox, that it still met your needs for your backbone panel.
Yeah, and don't assume just because you've always used SK1 that it's going to perform well. The best example I can think of this is BUV395. If you look at the SK1 versus the RPA-T8 conjugate of that, you'll find that the RPA-T8 conjugate performs extremely well as BUV395, but not SK1. Yes. And so you have to look at conjugate specificity when you're looking at a given clone. And not assume that one will always be the best. I mean, if you look at the BUVs and the BVs, they use a lot of M5E2 for CD14, whereas there's a lot of other clones you could have used, but that one just works best with those particular fluorochromes.
We have a question from the audience.
Hello, everyone. I have a question that how do you exclude the bias that is contributed by the observer towards data analysis? In the beginning of Kathleen's talk, she has mentioned that compensation can be tweaked by an eye, but I wanted to understand with respect to placing of the gates, demarcating the negative and the positive population, how do an observer be cautious about placing the right gate when we have a stretch of population that is towards left and right side, and we don't know that where to place the gate which would demarcate the positive from the negative population? So I think we see a lot of plots in Stephanie's presentation in which we have a span of population from negative to positive. So how we make sure that we are analyzing the data correctly and not biased with respect to every observer that analyzes it. Thank you.
Well, the answer is you need a baseline, right? And what's your baseline? So the truth is you shouldn't tweak or manually compensate a panel afterwards. It's just, you know, a Q-squared, it was not allowed. It was designed right. You made sure you didn't have these problems and you didn't have to adjust anything afterwards because so much of it is subjective. If you're forced into that, what you need to do is include an FMO for anything you may need an adjustment and make sure that, you know, that you're able to look at the X and the Y means and the Y and the X means, right? And make sure that the centers of density are lined up with a negative and make sure you actually have a negative to work with. So if you know you're going to have a problem in a panel and you can't avoid it, you need to add an FMO so you can check the compensation and have that baseline of that FMO. It's the only way to have it reliable. Otherwise, you're just flying by the seat of your pants, and you're right, it's completely subjective and not a good way to work.
Another way to put a control in is if, for example, in activated cells, if you could use an unactivated, then you are able to gate. That is ideal because you already are including the fluorochrome. So any background staining that might pull it up a little, it will be accounted for. FMOs are great when that's all you have, but technically without that fluorochrome in there, there could be a little bit of space, but it's really all you have to be unbiased, like the best you can.
And the other thing about activated cells is you should always keep some frozen in your freezer, 328 activation. So a Q-squared we made a lot and distributed across the 7 global laboratories. So when we were doing assay development, one of the steps was always to check for the markers where you needed to see activation to know if it was performing correctly. So a lot of people design panels and don't ever really check the activation markers or markers that are expressed under differential conditions. So make sure you always have some of those handy. Unfortunately, people have been trying to sell those, but they don't perform as well as the ones you make yourself for some strange reason. And you really should have those on hand if you're doing assay development.
Yeah, so I think Kathy has a question that's a bit related to this. Do you factor into this backbone panel suggested controls to use? If it is a specific tissue, do you test beads, cells for compensation, and guide users accordingly?
So we go with the principle of always using cells, but you could use beads for compensation, but then we typically run cells anyway, just to check and make sure that the performance was as expected. That way, if you forgot to add the antibody or just something happens, you have a second set of controls. But you could use beads for the compensation. There was another part to that question.
Do you factor into this backbone panel suggested controls to use?
Controls. Yes, well, that would be application specific. So the backbone panel was really just to get you to the panel. And then as you design how you're going to use that panel in your application, then yes, you need to have the appropriate controls, such as potential gating FMOs and the unstimulated controls, as we were just talking about.
The other thing you want to do is you can use a mixture of beads and cells. And so you'll look for it. There's 2 things you need to have a good compensation or unmixing control. You have to have something as bright as what you're going to be unmixing or compensating. And then you have to make sure you have a spectral match. So you need to look at the beads and the cells, run them at the same time as your controls, go back and look at the spectrum of them, make sure they're a spectral match, and make sure what you're using is bright enough. Sometimes it's really useful to use a surrogate. CD4 can be a good surrogate. CD45, if you drop in a negative, can be a really good surrogate, or use a single control in your unmixing and for your unmixing. But even in traditional flow cytometry, you should always make sure you have a spectral and a brightness match. It makes a big difference. A lot of the things we used to see in 2, 3, 4, 5, 8, 9, 10 color, we saw some problems where we didn't have correct compensation. For instance, if you look at an MDSC panel and you're running 14 and 15 in that panel, and you look at them juxtaposed against each other, you'll see that they're not really compensating correctly, because more often than not, people just use beads to compensate those. And when you're using the granulocytes and monocytes, you're usually, but not always, better off with a cellular control. So there's things where you might want to use a mixture of those.
Right. Okay, next question is for myself. What experience do I have with backbone panels? So I have briefly said, I've been working with Cytec a lot last year, and I have used their 25 color immunoprofiling kit that I've extended to a 32 color panel. Our challenge there was that we were working on a very specific clinical sample type, or maybe not, maybe people know it, but we are working with smart tubes, which represent freshly fixed whole blood. It makes it incredibly challenging, honestly. There is lots of variability in smart tubes collecting across clinical sites, because this was our application. We were looking at clinical samples collected across dozens of clinical sites. For example, we noticed during this work that the CD25 clone that was included in the Cytec backbone panel did not perform well on fixed samples. So that's part, there was a question earlier about checking how clones work on your sample, where this was, for example, an example where we had to change that. We had to adjust, a few staining conditions for CCR7 that, again, on fixed samples does not perform as well as in cryopreserved PBMC or something else. So I hope that answers the questions, but it was definitely very interesting work, and I can really vouch from a user perspective how important it is to have very well-built backbone panels that really sped up our development time for the panels that we in the end needed. In about 1 month, we had a 32 color panel, perfectly functioning, available, set up on our Cytec Aurora. And we've used it now to profile over 200 clinical samples with incredible reproducibility, other than varying quality in the samples themselves. But yes. And I think we are reaching the end of our session.
I think so. Thank you so much, Celine, for handling the roundtable, and thank you all for your talks and your participation. Right now, we are going to take a break from our YouTube, and everyone in person is going to go and have a little lunch and meet with some vendors. So thank you all, and hope to see you back. We'll start back up in an hour or at 1 o'clock for our next session.
Thank you.
Thank you.
All right. Bye-bye.
Okay, we're going to get started in just a few minutes, maybe 2 or 3 minutes.
We are going to continue in just few minutes.